Okay, good morning. Welcome to our second day of this symposium. Just to remind you, we had some changes in the program due to a few colleagues that couldn't make it to Montpellier and will present virtually from the United States of America. So we moved them to the afternoon in the symposium so that they don't need to wake up at 3 a.m. and talk to us. So now, in the beginning of the symposium, we will have a session, which is now session 4, and vector control and features modulating behavior. After that, we will have the session after coffee break. We will have a session dedicated to male behavior, and then after lunch, we will have the the session originally programmed for the starting in the morning, which is the session dedicated to repellents. And finally, we will have a session, the last session of the meeting, a session dedicated to oviposition behavior. And we will close the meeting with a presentation by Agenor Mafra-Neto from ISCA Technologies. So he will present kind of an experience of the corporate world, and there will be a final debate on how to frame these perspectives we share during the symposium in a broader context, and specifically in the One Health perspective. So I let the floor to Nadia, who will chair the first session today. Thank you.
Okay, good morning, everyone. Our first speaker today will be Carlo from IRD France, presenting novel quantitative approaches in the study of mosquito-periodic behaviors. Welcome.
Thank you very much. Thanks to the organizers for having invited me to this very nice symposium. So, the title is Novel Quantitative Approaches in the Study of Mosquito-Periodic Behaviors. There's a little bit of a word of caution that I have to say at the outset. It's about the novel. There's actually not anything really novel in what I will be presenting. It's something that biostatisticians know for ages, but what is perhaps novel is how we apply what I will be presenting the study of mosquito and vector periodic behaviors. So let me start with, first of all, the original and very famous KIM-RIVE 4 types of questions about behavior, about the origin of behavior, and the dichotomy about why questions versus how questions, and about how questions that are really basically concerned about mechanisms, how certain response, what has caused a certain response, what is the mechanism underlying it, compared to why questions, which we are more interested about the function and the adaptive value of a certain response and of a certain behavior. And so what I will be presenting can be applied, of course, to all types of questions here, but my main interest is, of course, in the why questions, in what are the functions and the adaptive values of behavior. And when we talk about adaptation, we talk about evolutionary biology, and evolutionary changes in behavior. So that's what I will be maybe concerned with in this talk. Why it is important to study evolution of behavior and adaptations in behavior. And I've just given here one example, but there could be many more. And it is about what would be called contemporary evolution. What is happening with respect to the biting behavior of anophelids? Most of you, of course, know that current mainstay controlled interventions against malaria rely upon insecticide-treated nets, which target the nocturnal feeding behavior of anophelids to protect people and to kill the vectors when they come to bite. And there is mounting evidence, and I've cited here one example from one of my colleagues, Nicoletta, who has shown that for one particular species, Anopheles domesticus, after 2 rounds of massive distribution of ITNs, he observed change in the biting time of Anopheles domesticus. And the question is, what is it happening here? Or is it ITNs that are changing this behavior? What are the selective pressures that are causing this? These are, of course, very important questions with very applied consequences. And when we study evolution, we are, of course, mainly concerned with adaptive, heritable phenotypic traits. And the main fuel of evolution is variability in a certain trait that is filtered by natural selection if it's adaptive. So I've taken here the evolution of the Apple logo, not the fruit, the company, through time, to give an example of certain problems that we are faced with when we try to study evolution of, in general, and evolution of behavior in particular. In this particular case, we could say, what are the traits that have evolved through time? And we could say, well, for example, the outline shape hasn't changed much, whereas the color has changed a lot. So color could be a measurable trait that we could follow up. But on the other hand, there are certain subtleties in color, for example, that are very difficult to quantify and to measure with. Let's say simple endpoints. And moreover, of course, this evolution here has followed some underlying causes that are not visible through the phenotype that we are observing. And these are concerned with the marketing strategies of the company and what were, for example, the product lines that they were trying to push forward. We cannot see that from these phenotypes. So there is a hidden layer here. And the same happens with behavior. We are usually concerned with what happens here. We are measuring responses. But responses are the outcome of a very complex network of interacting compartments and integration of different cues, for example, or endogenous states mediated by hormones, for example, and everything is filtered at certain points. For example, exogenous cues are filtered through environmental modulators that are filtered through endogenous modulators that can filter sensory receptors, that can filter perception, and everything is integrated at the central nervous system, which in return can define certain behavioral states, what I call here motivational states, that provide a certain probability of our response. And the probability is not is all or none, but we don't see what's happening behind the line. So we get a response, but we may not get the response, and everything is fed back into this loop. And there's the experience also that feedback into everything. Everything is very complex, and everything is happening behind the curtains. We are observing what happens here. We are not observing what happens behind the curtains. Just to give an example, we cannot really define sleep based on the absence of movement. So the response is movement, but we do not observe a state, which is sleep. The other thing that was in the title is periodic. So why periodic? Well, I'm very concerned with the behavior of mosquitoes, and almost everything in the life cycle of mosquitoes is periodic. If we accept perhaps the emergence of an adult from the larval state, most of the behaviors and behavioral events that happen throughout the life of a mosquito happens more than once and with certain periods and at certain frequencies that are characteristic of gender, of the species, and so forth. And all these are modulated by the environment. And within a certain cycle of 24 hours, for example, things happen not randomly, but they are structured and they happen synchronously at certain times. The other thing is about behavior, its dichotomous nature. It's like light in a sense. And I've used this analogy of galaxies to show you what I mean by that. So galaxies are very complex structures, but if we look at them globally, we see that there is order, there is a structure. But as soon as we zoom in, then order disappears, and we are confronted with chaos and randomness. And behavior is almost like that. If we see it at a global level, it's highly structured, and if we look at it at a very fine and local level, it's more random. So to kind of represent all these complexities, I've used some graphical paradigm here that are called lifelines. A lifeline is just a line that represents the flow of time through the life of an individual. And so there are endpoints at the end, at the beginning and at the end. This could be, for example, the emergence of an adult mosquitoes from the pupa, and this one could be, for example, its death. And through the lifeline, there is a sequence. The individual goes through a sequence of states, different states. Here is a representation of a binary situation, so two different states. And usually these states can be ordered in a very structured way. So, for example, if we say these are the alternance between activity and inactivity, we know, for example, that these are regulated by the biological clocks, and biological clocks operate with very defined frequencies. And here is the period, for example, with a single tau. And so we can kind of model and figure out this order structure through, for example, deterministic functions like a sinusoidal that represent the probability of being in each state through time. But within certain states, there are behavioral events happening, and these are more of a random nature. So they have kind of at certain point of time within a state, but their sequence is more random. So we can represent this, for example, as a random process that happens with a certain probability that can be modeled through probability distribution functions. Now that we've looked at all these characteristics, I'm going to present what we've been using to kind of use all this information and put it in an analytical framework that can handle all the complexities that I've just outlined. And these are hidden Markov models. I have no time, of course, to go into all the details, but very briefly, the lifeline here, and which is the flow of time and with the alternance of different behavioral states can be modeled through a Markov chain. Here, for example, we have a Markov chain of two states. Again, if I use the analogy of activity versus inactivity. And the change between states is modeled through probability of transition probabilities, probability to be in the same state, like these ones, or to move from one state to another one. And this can be modeled through what is called a transition matrix, with these parameters that are these probabilities here. And this is the hidden layer that we do not see. What we see is the process that we can observe. Like, for example, is the animal moving or is not moving? And the probability with which the process is observed at any point in time is determined by the state, the underlying state, and the random process. In this particular example, is like a simple Poisson process with a single parameter lambda, which provide the degree, the rate at which this process is expressed, and we can observe it in the hidden macro, in the as the response that we are looking at. So we've been using this kind of approach to study locomotor activity of anophelins, either in the lab or even in the field. And just to give you an example of the kind of results that we can and outcomes that we can have from this type of analytical approach. We have, for example, partitioned the sequence of beam breaks. We are using the kinetics locomotory activity monitor as our automatic recording system. So we can kind of partition this sequence into a sequence of different behavioral states, each one with its own movement parameter, and then model what is the probability of being in each particular state through time. And so in this way, we study the chronobiology, in a sense, of these behavioral states rather than the response itself. Okay, so let's move on. So far, we have looked at the lifelines as a 2D representation of the life, of the trajectory of a single individual. But in reality, what is actually a more a truer representation of the lifeline is not a 2D representation, but rather a circular, linear, 3D, three-dimensional representation, like the one you see here. Because everything goes in circle, and each circle we could, for example, assimilate it to the course of one day of 24 hours. So every loop here is a single day. And I'll try to do this one if I succeed. Yeah. If we try to look at the circular linear representation of the lifeline from the top, it's actually circular, of course. And we can try to compress it like in 2D rather than 3D and see how the events we are interested in can be can distribute around this circle rather than, as we saw before, the linear representation. And this is the domain that in statistics is called circular statistics or directional statistics. And there are useful ways to summarize this type of data. For example, in this particular case, this vector here represents the mean, the center of mass of the distribution of these events around the 24-hour clock. And the length, so this is pointing, for example, towards midnight. And the length of this vector represents how the degree of concentration of these events around the mean direction. So this is kind of neat way to summarize and represent how periodic behaviors around the 24 hours distribute and can be summarized and represented in this neat way. And we can also move on. So far, we've looked at the lifeline of a single individual, but we could use this approach to actually study population of individuals, because in some circumstances, we don't have the luxury to follow the life of a single individual, but we can rather try to understand a lifeline through the study of a population of individuals by slicing up the three-dimensional lifeline in single 2D lifelines like this one. In this particular case, we have many individuals distributing around the clock, and we can use, in this case, circular probability distribution models, functions to model this distribution. In this particular case, for example, I've used the von Mises distribution, which is a two-parameter distribution, which is the analogous of the normal distribution on the linear scale. And the von Mises uses two parameters, the mu, and which represents the mean direction, in this case around midnight again, and kappa, which is the concentration parameter. So in this particular case, you see that the points are not very concentrated around the mean direction, so the length of the vector is very, very small. And in this, another example, we have that the concentration is much higher, as you see from the vector, and most of the events are concentrated around the mean. We've used this approach to study a particular behavior, which is the biting behavior of anophelines. And this is a study that was performed in the Central African Republic, in the capital Bangui, with colleagues from the Institut Pasteur there. And we have been able for the first time to show the full periodicity of the biting cycle, of the biting events for a population of different species of anophelines. And the main result here has been that, which is important from the applied perspective, is that we've shown that for most of the species that we studied, the mean direction, the mean biting time didn't change much, but according to whether the collections were made indoors versus outdoors, the dispersion of these events were very, very different. And for biting events happening indoors, we basically had a very dispersed distribution, which from the applied point of view, it is important because, of course, when using bed nets, people during these hours are not protected by the bed net, so they are exposed to the bites of the anophelines. The other interesting way how we can use this approach, this analytical approach, is that, of course, in many cases, probably, as you can see here, the empirical distributions of these points are not, they do not conform to very simple probability distribution models like the von Mises that we've seen before. And so we can try to partition the sample data, the empirical data, as they are, as if they were a mixture of different circular distribution models with different parameters. Like, for example, in this case, two von Mises with different dispersion parameters and the same location parameter. And that's exactly what we've been doing with data that I've just shown. And we have come out with these particular results in which, in Bangui at least, we are perhaps confronted with the presence of different population of biting events, each one with its own particular mean direction and dispersion. And for the indoor biting, for example, the main result is that we probably have three populations, two that are biting early on or later on during the night for each species, and third population of biting events that is the one that is basically biting during the day. we still don't know what's the biological meaning of all this. But of course, this is very important to understand, and that's what we are working on at the moment. So that's the end, and of course, all this work has been collaborative work. So I want to acknowledge the help of all my colleagues at MiVEGEC, in particular Angelique Porchani, who has worked a lot on the hidden Markov models under the supervision of Raymondine and Nicolas Moirou, and Amadou Traoré, who has provided most of the locomotor activity data. Diego Yala for the work in the Central African Republic, and the colleagues at the Institute Cluster in Bangui. Claire Sangbake-Ngumu and Karine Gouani, and the funders, the French pioneer or founder of the proposal. Thank you.
Thank you so much. Any questions?
Very interesting. So I was wondering, so you share results with different parameters. It's very technical question actually. Did you do some multivariate analysis, or each one was tested independently?
No. For the mixture models, maybe?
Yes.
No, it's not tested independently. It's tested with all the data. So we are just modeling the mixtures,
Okay.
and we are estimating the parameters with an EM algorithm.
Okay.
How did you find this spread over three groups? And do you think it's a learning? Is it genetic? You try to see if you belong to that group.
As I said, we don't know yet. We don't know yet. We suspect that there is a great influence by the environment. There may be the presence of individuals that do not show rhythmic behavior, which may have been selected. So usually in a population, you may have some arrhythmic individuals, and these are very few usually. And maybe under these circumstances, they have been some way not selected against, and so they show up in your.
So one mosquito, one night could be concentrated biting, and another night could be right.
Not necessarily, no. It's not like that.
Not necessarily.
This is, as I said, what we are looking here. It's a population. So.
Yes, very nice talk. Thanks a lot. I wonder whether you sampled these mosquitoes to try to go to the black box and look at profiles in genetics, metabomics, anything. That's one question. And the other question is, are you trying to check whether these profiles, the diverse profiles, are repeatedly observed in the same individuals, or whether this is a thing that is not, let's say, an individual profile?
Yeah, the repeatability of behavior problem, of course. For some of the responses that we can measure, this could be done. Others probably couldn't. And what we are also working on, what we are aiming to do, is to also look whether there is some transgenerational effect, and so looking at some genetic behind what we are observing. Yes.
One more question.
Yeah, outdoors, of course, there are less people at night and during the day. So are your data normalized on the basis of the number of people, or just the total number of bites independent of the number of people? In which case, the outdoor biting at night is even more pronounced than where you expected it.
Okay, let me explain this. We are not looking here at the number of bites. We are looking here at the rhythmicity of the bites. So it doesn't matter how many bites you have. You are looking at how they distribute around 24 hours. So the question of how many people are inside or outside is another question. That's not what we are focusing on.
Thank you so much.
Thank you.
Okay, our next speaker is Aman Bonaventure Omondi from Benin, and he's going to be talking about interaction between fungi top disease and the vector banana aphids.
Thank you very much. First of all, the organizers of this meeting, coming to Korea, usually connect to the jobs. Our headquarters is here. I've been here many times in my past life, in my present, and hopefully also in my future. I would like to talk about host interactions in banana disease management, but as opposed to the main direction of talks of trying to generate address very fundamental questions, we are actually addressing the last line questions. Do we have effective ways of addressing a pest problem? But is it useful? When a method for addressing a problem is extremely effective, but requires a lot of investment or becomes difficult to implement, it is not very different from a method that is not effective, but that is used by a lot of people. So what we are trying to do then is digging back from the past of what has already been done and trying to see, is it working, is it popular, is it useful, and can it be scaled? Now, just in case I escape this, we have been funded through time from the CG, the CG core, and we also have bilateral projects with the University of Queensland that supported the work that I'm going to present here. And really, although I'm the one talking, this is the team that collected the data, and we work in collaboration in a network with partners in Africa, but also elsewhere. I've never met in my life anybody who did not know banana. What I've met, though, is a person who did not consume it correctly. And in Africa, as you move from east to west, people consume banana in very many different ways. But in Europe and most of Americans, I think it's just a sweet fruit. Now, so the processed material is, most of these are really dependent on the different pairs you're looking at. For example, matoke in Uganda or boiled banana in Burundi, different uses require different varieties, but also require different ways of managing the farms. If you look at the wine or the beer bananas, for example, they are planted and left probably to the base of God, and then they go back to harvest. It's a very low intensity system. While looking at the more cooking or dessert bananas that you have here, there's a little bit more intensity. Now, this is important to us because it influences the way in which diseases run across the landscape. Now, but we also have cultural uses that really don't care about whether the plant is sick or not. What I've also found out is that it's quite common, many of these flowers are quite common, and related to the banana, we have a lot of ornamental plants. We have some crops that tend to share a number of pests with the banana, and that is part of the question that we are trying to, that we are going to try to answer here. So, in case I do not complete this talk, all I wanted to say was, what is the influence of the status of the host with regards to the virus or some other stress on the population consequence or the fitness consequence of the vector, and what epidemiological consequence does that have? But first, let's talk about the banana biting disease it's not one that you find many people have seen before. The bunch top disease shows up something like this. The banana looks like a bunch. Why? Because when a banana plant is growing, the younger leaves tend to be bigger than the older ones. The bunch top disease just reverses that, and so it becomes kind of like a bunch of flowers. Now, and the true stem of this plant is actually the same plant, or let's say the same match, and this true stem is underground. All that is up there are leaves. For banana bunch top disease, as opposed to other diseases, the two are connected such that if this seems sick, that is surely sick as well. So it cannot be a source of seed, for example. But while everybody can see that, the point where control will be effective is at the beginning of the infection or development of symptoms, where action can be taken to save the plantation. Surely that will not save the plant because the plants don't heal. And the vector, the Apterus type, is a Pentalonia nigronegosa, the banana aphid, which for a long time has been assumed to be only restricted to banana. Now, where do we find bunch top disease in the world? It's everywhere banana is grown, except in Latin America. It's also in Africa, really spreading across the main banana growing regions. And so if you go by country, it's probably in 80% of banana growing countries. But what strikes us is that while the first introduction was in Egypt about the turn of the last century, and in Congo about 1950s, it had remained in Congo for a while, probably reduced canopy connectivity and movement of people with seed. But in the recent past, last year, there were 2 detections in Uganda and Tanzania, first reports, 2019 in Mozambique and 2020 in Togo. So there seems to be a faster and faster spread of the disease in Africa. I've already spoken a little bit about this. When we look at the banana bunch top disease, we actually go from the cigar leaf as we go down. When suppose this plant was infected at leaf that we call here 3, the leaf 3 may not show symptoms, and it's only leaf 1 that will show symptoms, so that the older leaves usually remain normal. They usually remain looking healthy, but the younger leaves show greater and greater symptom expression as the disease progresses. And here a chain of earlier symptoms that we can see, and the later symptoms that most people really detect. If we were able to have many producers detecting this range of symptoms consistently and then taking action, control is actually possible. The Australians already showed this early, about 1924. And how do we work? Now, we are not trying to eliminate banana bunch top disease. We realize we probably don't have the capacity to. We are trying to recover production, even though there might be bunch top disease somewhere in the landscape, and to expand the production, sustainable production area, so that eventually we'll be able to recover large areas where production will be possible, even though there is bunch top in those regions. And that is heavily based on reducing vector access to inoculum, and also getting clean seed, and therefore maintaining so that the first clean seed becomes a secondary source of low cost, low risk seed that would eventually give production. And every pilot site is manned by an institution in a different country. And so within those countries, what they have to do is to set their own priorities, set how they will achieve those priorities, and report back on what works and what didn't work. Now, as COVID has shown us, where you have a very scarce resource, the big boys eat first, but that doesn't mean that they will remain safe. So within the villages, what we see is that as long as we don't have a way of compelling compliance and getting seed so that we really minimize vector contact with inoculum, the disease surely comes back. So what we tried to do then was to apply the Australian protocol by training farmers on what to do. The assumption has been that only trained technicians would be able to detect the early symptoms, but now we wanted to try it with farmers. We don't know how much they learned. What we know is that we provided them with a capacity to detect, and some obviously were able to identify the disease. We also provided a social supervision system where a committee, every farmer here, different farms, and here weeks in a month, every farmer would monitor their own farm once a month. At the same time, a committee from among themselves would monitor randomly the different farms and bring back a report, because the committee would then report to us on the number of plants removed in that month. At the same time, the extension personnel would then follow up and supervise the committee. So we have knowledge, but we also have compelling compliance. What we found in this experiment that began in 2016, August 2016, by 2020, they had maintained a very decent level of banana bunch prevalence, way below 2%, and they are supporting production and expanding. But the first year of this experiment, in truth, really failed, because we got only totals of annual plant removal, and we set up parallel experiments, one in Burundi, in the farmers' fields, but supervised by our own technician, and one non-replicated experiment in Benin, where we wanted to know, suppose we inspected twice a month with every leaf that came out, would we be able to completely eliminate bunch top at a point, and how long would it take to come back after the last elimination? And what these point to is that, yes, there's a possibility of using this fairly low-tech but high-intensity approach. However, it wasn't all rosy. This is a non-controlled experiment that was just to look at the trajectory of infection set up in Malawi around the same time. And this is the level of reported detection of the disease that the farmers gave us. And that is their second seed farm, actually. But then we also had this group in Malawi. And their farms did not collapse, but it was not anything you wanted to show visitors. And what we see, we had thought that maybe they did not follow the protocols very well, and seemingly there is evidence about that. But the two groups around this area, is lowland next to Lake Malawi, seemed to achieve the same result. And so we went asking ourselves, is it possible that they just ran into a level of vector movement, a level of vector activity, a level of new infections that our approach could not stop? Or could there have been different factors that we could consider in ensuring that we don't have this, but eventually we have this kind of expansion? So, I will pause there for a bit, but I'll come back to those questions, and I will show you some work that we've been trying to do. We acquired a small UAV in 2016, and we were trying to use it to detect disease and coordinate farmer responses. However, this was probably one of the ways of pushing together collaboration, but also of supporting detection, we had hoped. But what we ended up doing was to model factors associated or determining infection rate in gardens. And after quite a bit of spatial analysis, what we found was that vector activity within infected field did not seem correlated with bunch top disease levels. But vector activity in the surrounding gardens seemed to be weakly correlated. What gave us a very important association was the banana density within 30 meters band and within 60 meters band. And we also, that was also quite linked to bunch of disease prevalence. Of course, we are working in endemic landscapes. And if the farmers had applied a banana-free fallow, or if they were cultivating in outside gardens rather than backyard gardens, they would have lower levels of infection. What we had not considered, and what came out a little bit surprising, was that where there were definite hedges around the gardens, then we had islands of fairly low infection. And so we've come back to ask vector-linked questions that relate to what we have seen. So we are setting up experiments that are pretty simple. We first of all tried to assess among the plants that we find, the species that we find around these beds, which ones may serve as alternative hosts or as refuge that could explain or could enable create bridges for infection to move from one garden to another garden. Because our banana-free fallow is focusing on banana, with the previous realization that it is known now that we have two species here, and one prefers banana and the other prefers other members of the family. Now, so when we did these experiments, we were able to have some level of preference, and we also tried to have a no-choice experiment on bananas, but this time looking at infected and healthy plants. Now, what we found out for the first experiment was that it was not true that other Zingiberales plants like ginger, like ornamentals, were really not preferred by the banana aphid, Pentallonia nigronevosa. Sometimes there was equal preference, but sometimes there was preference for banana. And in this case, we have many of these plants as urban ornamentals. This reminds me of greening cities. We have a lot of bananas showing up in backyards, especially in Africa. And other than those, we also have many of these ornamentals that are generally lower canopy, tolerant to shade, but could mean something in vegetal disease control. And then we tried to test whether there was a preference for infected and non-infected plants, diseased and non-diseased plants. But what we find out here was a little bit telling, that when the aphids were not carrying the virus, aphids that had been raised from healthy plants seemed to prefer infected banana plants. Very weak preference, but significant preference. But when we used aphids that had been feeding on infected plants, then this preference we saw here is lost. With aphids, we say viruliferas are not infected, and we didn't expect to have the virus to affect the aphids directly. But we can say, well, a few questions we would like to ask. What is happening when the viruses, when the aphids are carrying the virus? And what is happening between the plants, the hosts that we are comparing? Probably is a presentation of some opportunity, probably is a way that enables the virus to spread faster. So we did very basic life table studies to compare aphids on infected plants and aphids on non-infected plants. But what we see is that when we raise the aphids on diseased plants, they were reproductive for a longer time. They started reproducing earlier and kept reproducing for a longer time. And the survival for the first repeat, for the first season we did the experiment, there was a significant better survival of aphids on diseased plants. But in the second season we tried this, that did not seem to be repeatable. However, these other results were repeatable, and also we saw that the age-specific reproductive rate also seemed to be quite repeatable between the two, though the magnitude of the difference is lower. And so we tried to ask the first questions. Does bunch top disease influence the nutritional quality of the plant? Looking at crude protein content as a signal, no. Looking at lipid content as a signal, also no. And looking at ratios between them, also no. But looking at the variability, we think that we could do something else, and we could probably look a little bit more closely at host quality, probably host quality 2.0, where I would want to see if there are the simpler sugars that could probably influence feeding behavior that could result in the consequences we are seeing, but for the moment we don't have this data yet. A summary of the data I've just presented here was that there was a consistent increase in productive rate, net productive rate. There's a consistent increase in decrease in generation time, and essentially aphids on infected plants did better and had a fitness advantage over those on non-infected plants. So some reflections. Yes, diseased plantlets were offered better fitness for vectors. And this would tell us then that where we have aerobic activity and infection rates are rising very fast, we probably may not succeed much. We have not tested this in the field. We just think so, and we would like to test this beginning this year. We did not see any change in host quality, and we do not know under what mechanism the result we are getting comes about. There's a possibility that when the host is stressed anyway, the vectors will perform better. And that stress doesn't necessarily have to be a bunch of disease. And so we are moving ahead to try and test this. We are trying to look at the actual feeding behavior to see whether there will be greater feeding or there will be a change in feeding behavior so that quality alone would not explain what we see. And we would like to look at refine our host quality assessment and look at epidemiological consequences. Now, in the last 2 minutes, I will talk about some work that we are just beginning. We were interested in the long-range attraction, and we have been beginning with headspace collections and doing the very fast analysis. All I want to say here is that we see that infected plants have a greater overall yield of volatiles that are coming from them, but also we see around there that we have also differences in the kinds of volatiles that are enhanced. But we do not know yet what that means, and that is what I would like to continue with. Now, and then finally, a student that thought there was a very different reason to performance of our logging activity. Remember, we are trying to see, can it work? Came in to try and look at the cultivar differences, and if that would relate to performance of logging as a way of managing a bunch of disease in different parts of the continent. However, he began with the lockdowns of COVID, and so he modified his work so that he wanted to ask also healthy individuals that are susceptible, once exposed, at what point do they get diseased, at what point do they get infectious. For bunch of disease, there is no healing, so there is no immunity, and the plants really don't die. The plants remain on the garden for a long time, usually packing very, very many aphids. And in brief, what he did was to infect batches of plants and monitor them over time until the symptoms emerged. And each time he tried to assess the early symptoms and the late symptoms emerging. And then he also clipped aphids onto them to have access to the plant for about 48 hours and transferred them to plantlets of a single variety to see whether those plants would eventually develop the symptoms. Now, in brief, what he found was interesting, that we used 3 varieties. 2 of these are plantain In West Africa, you must specify this, but these two, there's a hybrid banana and there is a sweet banana variety. Now, the hybrid came up first, going through all the stages of the disease, developing early symptoms to late symptoms. But the landrace first looked like it was never going to develop any symptoms, and then came up with the final level symptoms, skipping the early symptoms and came up with the final advanced symptoms after about 15, 16 weeks. Now, if you have plants like this in the garden, are they infectious? And that got us worrying a little bit. But what we found out later was that actually it was the onset of the first symptom that was associated with the infectiousness, whether it was this one or it was this one. So these only became infectious at this point, and these only became infectious at this point. We do not think this will hold for a long time. There might be cultivars that are infectious all through, and if that happens, then our hope crashes in roguing as symptomatic roguing as a way of eliminating disease. But also, the success of infection differed between the different cultivars, the hybrid being quite different from the local landrace. But the two plantains were somewhere in between. Our colleagues from Cambridge had actually done some modeling to see whether knowledge and frequency would compensate each other using our data from Malawi and using also data from other pilot sites, looking at how frequently farmers were capable of doing budget disease control activities. And what they say, well, if you don't know, you better do it more often, and then you will still be able to achieve some level of control. The red are where the population crashes completely, and so you can no longer have production after that point. And the blue are where you can really keep the garden going. So, what we have done and what we are doing, we are trying to refine parameter estimates that our modeling colleagues will use, and hopefully we will be able to get a much better performance environment mapping of bunch-top infection. We are trying to look at the possibility of canopy mixtures or crop mixtures, and we are also trying to refine our scouting protocols to see whether these will be able to perform much better. But most importantly, there seems to be a seasonal stress that many plants go through in a year, and potentially this could provide weak points or opportunities for bunch-top disease. Thank you again for inviting me here. If I come again, these are the two new students that are working on the other stressors, whether AI-aided detection would really perform better with the farmers, or whether there's a seasonal variation in disease weakness or potential to become infectious by the banana plants. And you have already seen the rest of them. We got our funding from the University of Queensland and from the city. Thank you.
So much. Any questions? Perfect. Yes.
Hi, Aman, thank you for this presentation. It was very, very nice. I was wondering, these differences defined in life-history traits, those it's hitting on the text in terms of how they're looking for differences in using content and so on. Have you looked at differences, at the difference in farms that in infected farms might be lacking because of the trade-off of the I mean, if I remember well, there was a trade-off between sickness, virus infections, and so on, and a vigorous compounds detected against the virus compounds. Perhaps you find some differences there.
Yes. The simple answer is no, we have not, but we would like to. And for our mandate, some of these studies are sometimes get a little bit difficult to justify. I would really love to have a look. And this is why we are going behind the door to try and stress the plan some other way, because then we can have a better justification to now go straight to look at the formats. Because most of the time it is, does it relate to control? Can it be scaled? So yes, that would be something very useful to look at. if the host defense is playing a role directly. Yeah.
Thanks, Aman. Very nice story. Wondering, you showed that the infected plants have a different volatile profile, and that seems to connect to the healthy aphids preferring them. Do you think you can use them as sinks for, use them, their attractivity to induce control on them?
It would be interesting if we would. It would be very interesting. Up to now, we do not know the behavioral consequences of these volatiles. This is something that we really like to study so that we remove the presence of the plant that has tactile whatever it is, and also has audio signal from the actual pure audio signal. But we would also like to have a look at some of the closely related plant species or some cultivars. Can we find ways of getting aphids from the more precious stands? We do not know yet, but it's something we like to do.
Do they have no predators?
They do have predators. They do have predators, they do have parasitoids, and my colleagues are working on pathogens as well.
I didn't catch early on, but is this a persistent or a non-persistent virus?
It's persistent.
Okay.
So once the aphids are infected, they remain infected for life.
Okay. Do you know whether it's getting moved more by the A-late or the actors? Is it getting jumped into new fields or is it spreading to neighbours?
Reasonably, movement between field to field is by elephants. But the apterals are also moved around quite a bit within the field, and especially in nurseries by ants. There's a huge association between ants and the apterals.
The ants are farming.
But the ants are tending them and moving them around.
Okay, because that causes a big problem in one of the pineapple viruses. I can't remember which one was in the land. So you could, I just thought it was very patched when you showed it. It was very patched. Okay.
I think we do not have right, our final speaker for this section.
Very much to organizers for organizing this very excellent for inviting me. I would like to talk about some limiting factors of population growth of Hematophilus mite with a focus on spatial configuration at the mite scale and relation processes to stress the need for basic research on tiny animals to advance pest control. Investigating population dynamics of tiny arthropods in agroecosystems, whether livestock or crops, is a challenge because there is a diversity of impediments. I will talk about a bit next. And the problem is it's very important, mainly because understanding the population dynamics of those tiny arthropods, those tiny pest arthropods, is related to major issues of integrated pest management. First of all, because there is a need usually to set economic threshold to optimize treatment type in IPM and to evaluate the regulating power of natural enemies on pest populations. The main tools, the most commonly used tools are in vitro life cycle bioassays, field data, but with field data we need to be highly cautious, especially with the model I will talk about, because with small arthropods, especially with Hematophilus arthropods, the pest population size is very difficult to estimate in the field because of the mostly cluster distribution. And we also can use mathematical models to refine how treatment could be optimized. But this is usually very hard to evaluate the realism of mathematical model predictions of population growth in tiny, in many arthropods, but especially in tiny hematophagous arthropods. So that's why I would like to tell part of an experimental story focused on a hematophagous mite that is a problem in poultry farms. So experimental story that generated important null results and allowed us to get answers to unasked questions. Because likely, as with some other tiny arthropods, but this is tricky, this is obvious here, with the Poultry Red Mite Dermanosus gallinae, that is a huge problem in the egg producing industry in Europe and worldwide. We are largely ignoring in terms of biology and population dynamics. This mite, the Poultry Red Mite, is red when it has been freshly fed with blood. It is an obligatory hematophagous mite that is parasitizing birds, and it is responsible for a diversity of damages in layer farms, both producing layer farms, but also it's also a problem in small hen houses in backyards, in family backyards. So it's generating diversity of health and welfare damages. It lives at a distance from the host, as do bedbugs or the female mosquitoes, but it's not winged as are bedbugs, so it accumulates in a diversity of cracks and crevices in the wall of the nest in the wild, but also in the very diverse cracks and crevices around hens in farms. And this, along with other features, makes it highly recalcitrant to conventionally sprayed acaricide treatments, because the population is something like an iceberg. Only the emerged part of which is touched by those sprays. Just to point on the model by Huber et al. 2011, this is a mathematical model that predicts population growth over a given time interval starting from a given number of mites of defined sexes and ages. This is a model, the parameters of which were at least partly drawn from literature, mainly from live tables that were established from lab experiments by several teams. So I will talk about this model because we've used it in the next experiments. That's why I pointed here. So the model is the model. For a long time, many people are trying to implement integrated pest management against DG. So Dermanosus gallinae, the Poultry Red Mite, will be called DG now. But to date, almost nobody did manage to have something satisfying. To develop integrated pest management, many different alternative management means to synthetic pesticides have been developed or are under development. The modes of actions are roughly listed here. But I have to say that to date, none are fully satisfying on farm. And we tested part of them, the gray highlighted ones, at lab and on farms with strong and important experimental work, with, to be honest, very informative results, but no results. So if we focus on biocontrol, biological control, that consists of making benefit of natural enemies, especially predators, to control the pest. The classical augmentative biocontrol, to date, didn't show any true effect, any regulating effect. So inoculating mass-reared natural enemies to date has not been satisfying. So I would like to focus here on conservation biocontrol that consists in favoring naturally occurring natural DG enemies. Just a point here, we didn't test those mechanical means, but you'll see that maybe they are much more interesting than we currently consider. These are mechanical means that are dedicated to obstructing paths to the host or to shelters. Electric perches are sold in some cases. Silica powder may help, and grease introduced in shelters, in mite shelters, seem to help a bit, but we didn't explore those very well to date. So let's focus on possibilities for conservation biological control. The question is, can we make benefit of predation processes between native mites to manage DG in poultry farms? Of course, there are methodological locks, and we managed to solve this by confronting diversity of different experimental and analytic means. The locks are mainly due to two main points. First of all, have in mind that it's impossible to observe directly predation events between mites, mainly because mites are small, but also, and first because they are living in cracks and crevices and in porous substrates such as dust and manure. Second, but this is a general rule, it's very hard to predict the regulating effect of one population, predator population, on one pest or prey population in a given ecosystem because of the biotic network that may strongly blur the issue. And I know that in the mind of many people, henhouse ecosystem, because it's important to have in mind that DG doesn't live on host, but you only can find it in the farm, in the building, something. You cannot find them on the outside area, even though it's free range. And this is the same in the henhouse, backyard henhouses. But if you consider the ecosystem within the henhouse, usually you can think it's very simple, not so many interactors, but you will see that there are much more interactors, even to just considering arthropods. So to make emerge the properties of arthropod communities, we decided to confront over several successive projects. Correlative analysis from field data, lab bioassays are at two scales, small case in vitro and medium scales with mesocostoms that allow to work on at the population level. And we also use the mathematical model by Huber et al. To make simulations and predict the population growth of DG. We followed, because as I said, we are largely ignoring a lot on biology of this mite, also economic importance. So we followed an approach with a sequence of prediction tests, testing hypotheses, then getting results, analyzing them, getting new information, including unexpected information, and then elaborating new hypotheses to test. Here are, so I would like to tell you part of the story with three small studies. The first one is large, but I will not detail it so much. I will pass very quickly on the first steps. But just have in mind the specific hypothesis we tested. First here, there are four hypotheses formulated as questions, because for conservation biocontrol to be efficient, first of all, obviously, there is a need for native predators to be present and, if possible, diverse in the hen houses. Second, out of these predators, we need to have predators able to feed on our target, DG. And last but not least, DG predator, so predator interactions involving DG should occur on field, on farm. And of course, because it seems to be the same, but this is not, you will see. We need also to check that one, if predator interactions occur, those interactions have a regulating effect on the DG population. So here is a very short summary of the first the experiments for to answer the first 3 questions. We conducted some important field work followed by lab work on 20 barn hen houses over 4 seasons. We recorded arthropods DNA from airborne particles, and we also recorded morphospecies from manure from different standard samples. We also conducted in vitro predation bioassays at lab with the native predators. Yes, because we found native predators, they were diverse and abundant. You can see that roughly the structure, this is just a scheme, but we found the structure of a food web typical of bird nest, which is not so much surprising, with the bird providing blood and feces and feeding on materials that are of interest or diversity of ecological grids. And above these, predators may feed on those games. We found several dozens predator species, mainly mite species, but also a few pseudoscorpionids and beetles. There were also omnivorous animals, arthropods. I don't talk about other organisms, but this was even interesting. Amongst the diverse and abundant predators, 3 were able to feed on DG, and they were almost omnipresent. We also noticed that astigmatic mites, namely the dust mites, if you are allergic to mites, usually you are allergic to a few species of astigmata. So astigmata were omnipresent, absolutely omnipresent, extremely abundant on farms. So we'll consider them as an integral part of the ecosystem. The next steps. So roughly, the answers to the first 3 questions were very, very promising. There were lots of mites, lots of arthropods, including lots of predators, and 3 predatory taxa were able to feed on DG in vitro, so without any barrier. But also we detected cues of effective interactions, pre-predator interactions between those 3 taxa in the field. Okay, so now what about the regulating effect of those predators on the DG population? To test it, as I said, it's very hard to connect the different results and to handle population levels effects within an ecosystem. Therefore, we designed closed poultry mesocosms as a new tool to investigate tiny hematophagous arthropods. These are systems that allow to home a chick in a mite-proof system, and the chick is fed and watered at libitum from outside. Manure is allowed to accumulate as in farm, and etc. And of course, one experiment contains several different replicates in the mesocosms because there is a need to estimate the balance of population growth to draw conclusions. So one experiment was composed of 8 to 10 mesocosms per modality run together. One modality was either control or one test with one or several predators. We introduced one chick at TO plus the mite inoculum We maintain these over several dozens days in order to have multiple mite generations. Roughly, DG has we thought it was 1 week long as a generation. So then at the end of the experiment, usually several weeks, we recovered and counted the mites contained in all of the mesocosms. This is 40 centimeters in diameter and height. The mite inoculum was made of DG and astigmatic mites, the only present detectable test mites, and in all mesocosms, and the test contained also predatory mites. The results were obvious and quite null. I mean, in those experiments with the controlled presence and absence of predators in a farm-like ecosystem, allowed a pretty good population growth of predators, but those predators that were found to feed on DG in vitro and on the field didn't exert any regulating power. We don't know if top-down regulation is absent from the system or whether predation was diverted to astigmatic mites because they were present everywhere, as I said, on farm, or whether there is any other explanation. But those two experiments, 5 to 7 weeks long, with different inocula at start and quite high predator to prey ratio, show that it's not efficient, and this is very consistent with what we found with mass-related inoculations. We found an unexpected information, by the way, along the way, because the measured population growth in control was far higher than predicted by the Uberta model. We were using this model to design the experiment, and we expected to have around 1,000 times less mites at the end, because at the end we got millions of mites when the starting inoculum was dozens or hundreds of mites. So this unexpected info led us to conduct another study to characterize the temporal dynamics of temporal population dynamics of DG. I will not detail the results, just to let you know that we used standard mesocosms with different intervals to measure the deviation from model predictions and to refine the understanding. And along the way, we found another unexpected information. The Germanistus gallinii population growth was drastically slowed down in mesocosms with hosts available intermittently and a constraint path to the host. This was due to constraints, experimental constraints, and I will detail it if I have time to, but just trust me. So when the mite had just a single path to reach the host, although it is very active and able to walk around the whole mesocosm within a few minutes, the population growth was strongly slowed down, but this was also in the presence of a chick available very intermittently. So we conducted a third study that allowed us to ask whether the slowing down, this drastic slowing down, was due to the intermittent availability of host or to the restricted path to the host. And apparently, this is due to the restricted path to the host. Maybe, I don't know if I have time to explain, but I will, just to show you, you can see that the mite is very quick. This is an electronic tracker, I will not detail, but just to reach conclusion, this is very funny because although it's unexpected, there is a funny convergence between current and our research, because Wood in 1917 wrote that about DG he wrote that the ease with which mice reach the host has a decided bearing on the rapidity of increase, it means population increase. And he recommended to conseil roosts that are not so much easy to be reached by the mite to control it. And that led us to better consider those mechanical means that we didn't explore to date, because likely it could help progressing the integrated pest management. So there are several conclusions related to DG management that are that we draw from unexpected results that allowed us to elaborate new hypotheses to test and so on with the sequence described previously. And I think it's very important to have in mind that it's very tricky to test the realism of model predictions. This is also why likely Abrams in 1992 was asking why in the literature we couldn't see so many cases where predators have positive effects on prey populations, in spite of many theoretical bases suggesting it. So I think that we really need to conduct more experiments, but that's very tricky and demanding. I would like to thank people involved in it, my previous PhD student, Sebastian Dupré, very good and Master 2 students, and Sophie Soudet and Lisa Dadu, who were engineer assistants, and Dominique Bicou, the designer of the Huber model, who is a very good collaborator, and we are progressing with him.
I think we have time for one or two questions before coffee break. One needs a bit of coffee. Yeah, Nicoletta.
Is there a non-aggregation pheromone or sex pheromones that's being used for attraction, managing the mite?
I am in the process of thinking about it. It was not chemically characterized, but an old study obviously shows it exists. And our last experiments, because I am working on attractant and re-emergence, obviously shows that the body of the mite is emitting something very attractive in different modalities. Especially, I think the most attracting is paper strips that are laid for a few days in mite aggregates.
So the chemical structure is unknown?
No, it's absolutely not known. We just know it exists.
Okay.
And I would like to work on it in the next future. I think that there are some work, some ongoing work on that.
Great, thank you so much.
Thank you very much. It's a pleasure to introduce it was Sharon here, who's supposed to be the chairman, and I don't know what she did yesterday night, she has no voice, so I will be doing my job. So it's a pleasure for me to introduce Nadia Melo, and she comes from Lund University, and I think she's got one of the best questions ever.
Yeah.
No
pressure. Right. I'm just gonna,
Yes.
if that's okay.
Thank you.
Oh, yeah.
Oh.
That's not showing on mine. It's on that one. Right, okay. So, understanding male, a new aspect in mosquito control. So this is a new project. We started with this project at the beginning of this year, and as the title says, we're working with males. And what we're trying to figure out, basically, is male behavior in regards to swarming and in regards to males and males. Okay, so the focus is not on the females this time. Right, so if we start with a little bit of background quickly. So what do we know when it comes to Aedes aegypti and swarming? Well, we know that they supposedly swarm around the human. We know that this usually happens onset photophase, and we know that males probably do it because they want to increase their chances of mating when the female approaches the host. We also know that in regards to Aedes aegypti, they don't swarm in large numbers like Anopheles. So it has even been suggested that once they reach a threshold, they go into these so-called macroswarms. Okay. In addition to that, since people are saying, well, they swarm around human hosts, the first thing that I did was to look at, are they attracted to humans? And according to current literature, males gain attraction to hosts after you knock out the fruitless gene, not before. However, a recent study that came out showed that males are attracted to humans at long range. So they managed to show this in videos, and they concluded that there is an attraction to the human host at long range, but not at short range. Okay, when it comes to pheromones and Aedes aegypti, it's even more wishy-washy. Basically, we have a few studies that have suggested that there are certain compounds that function as an aggregation pheromone. They have suggested that this aggregation pheromone is to attract females. There are some that attract males that females release, and some that are maybe both for females and males. So as I said, we don't really know what's going on. Okay? Right. So that leads to our question. And our question, as I said, is quite a recent project, so it's not really defined, but this is what we are basing ourselves on. So this is what we want to find out. So if you think about it as swarming, what happens is that they group. They do this to become more successful in mating. So the more they group, the more the advantages are. You know, you have increased chances of mating. So what happens? Is it that there are aggregation cues, or is it that host cues attract the males towards them and then initiates the swarming? Once they are in the swarm, well, where's the threshold? And when the threshold has been met, how do they know that it has been met? What happens once the threshold is met? Well, you usually see competition. You can see aggression between the males. You see this dispersal microswarm. And our question here is, well, do you think also that perhaps they are releasing chemical cues that are signaling that, okay, it's enough, we're too many, go away. So this is basically the idea behind the project. And I know that when it comes to swarming, people talk about sound, and that's important, but I'm gonna be focusing on olfaction, okay? Right. Okay. So the first thing that we did was, okay, how is swarming initiated? Let's work with human hosts, and let's work with males. So we did this uniport assay. Oh, sorry, the picture is a bit blurry. We did this uniport assay where basically you release one male at a time, and then you have your odor source here, which in our case was males or host. You have the airstream going downwards, and then if the mosquito leaves the release chamber into this big tube, it's considered activated. If it goes through to this tiny one and reaches the odor source, it's considered attracted. If it just stays there and does nothing, it's not activated. Okay, so that was the first thing that we did. And what we found is we measured in percentage attracted mosquitoes, and that control, of course, we didn't really see anything. Then when we tested with the hand, they weren't really attracted either. There was no difference between control and the human host hand. Then when we had other males in the containing chamber, suddenly we saw an attraction. It's not a very big attraction. That was a bit disappointing when I got these results. But then I thought, okay, so swarming supposedly occurs near the host and with other males. So what if I mix both males and hosts together in the holding chamber? And suddenly attraction went up to about 60%. So this was exciting. Okay, something is happening. There's a synergistic effect when you have the host and the males. There's something going on also when you only have the males, but not as strong as when both are present. Now, being interested in olfaction, I went on to chemical cues. So the first thing that we did was to collect odor from both males and females. We tested different types of collection. We did hit space, which is not very good. So then we ended up trying SPME. And what we found is that even though the profile doesn't differ very much between both genders, males tend to have a larger amount of this compound here. And this was repeated several times to see that it was actually there. And after a few repetitions, we saw that it was there. So it seems that males are constantly releasing a blend of lactone homologs. So we found this one that was the most abundant one, lactone 1, and then we just named the other ones 1, 2, 3, 4, 5. So now that we can confirm that this is present in the male sample and a higher abundance, we decided let's identify them and get the synthetic compound to see if something happens. So we started with the one that was most abundant, lactone 1. And what we could see is that yes, when we tested it, well, we put the compound here and released males, they were attracted. It was a higher attraction than when we tested only males in the previous behavioral assay. And then I thought, okay, what if I mix the lactone with my hand? Would it then be more attractive? And it was. It increases a bit, but not super much. Then I just wanted to compare it with the results that we had from previous one where we actually used the males and the hand. And it's pretty much the same. There's no difference. So far, we found something that is released by males and females that is attracting males. Then I thought, okay, well, let's check with females to see if it has the same effect. So it doesn't have any effect on females. Females don't really care. So I didn't test it with host hand because they would obviously be very attracted since you're putting in the host odor. Right, okay, so here we are with something that is specific to males. It's released by both, but it's only attracting males. Okay, so I started to think again, okay, well, what does this mean? And how come when the host odor is present, mosquitoes are more attracted? So basically, I went back to the question, okay, well, what is going on in relation to male odor or aggregation cues, we don't know, and host odor. So what I thought would be interesting to do next is to observe a little bit, let me see what's actually happening. So we recorded 3 different scenarios where we had basically just air, stimulus, and males inside. Then we had with hand, and same amount of males inside, and then the lactone 1 that was so attractive. And then I observed this for the same amount of time that I conduct the behavioral assays, just to have like a limitation. And it was very clear when we did this that something is going on. The males are much more active for a longer period of time when you have a host hand present. Lactone 1, on the other hand, didn't seem to do very much to the actual activity of the males. So we tried to put this into parameters. So this is basically what we could see, number of active mosquitoes approximately after each minute of the experiment. So this just confirmed the video where we can see that the presence of a host not only made it last longer, but they were much more active also when they were together with the hand. Lactone 1, on the other hand, didn't really have an effect on the swarming or on the duration of them being active. So far what we can conclude is that while lactone 1 is attractive on its own, it doesn't seem to either initiate warming, if we can call it that, or have an effect on the duration. Okay, so back to square one. And then we decided, well, let's collect odor of everything. So what you saw in the videos, let's collect odor and see what's going on. So that's what we did. We collected odor from basically the same assay that I recorded and tried to see if we find something interesting. And this is where we are at right now. But so far, these are the results. So what we found here is that there is a variation in the volatile profile emitted by these 3 different sources. But what first caught our attention was this one here, because that one was lactone 5. And that one was one of the least abundant ones when we collected in the beginning. So now suddenly when we have this environment where the males are very active, they are flying for much longer, the amount of this lactone that they are releasing is also significantly higher than to begin with. So we were very excited when we saw this. I thought, wow, maybe this is because this is what's attracting them more when you have both host and males. Okay, so we identified it, went through the whole GCMS story, and got the synthetic compound, and went back to the behavioral ones. It wasn't that great. So they are attracted, but not in the same intensity as the first lactone. Okay, so right, this is results that we got last week, and just in a rush trying to figure out what's going on here, when we were doing the assays, what we could see is that the males do not fly into the attracted area as much as when they are exposed to lactone 1, but they go crazy in here. So of course we are planning to track this because it's not easy to explain it by just showing graphs, but that's something that we observed. And when we looked instead of number of activated males, it reached 50%. And the funny part also is that males went into the attractive part, but came out again and kept flying around. So something is happening. Something is happening. What's happening? We don't know yet. So to summarize this, what do we know so far? What can we conclude so far? Well, males release a blend of lactones in higher abundance than females. At least one of these ones are causing an attraction on other males, not females. And the ratio of this blend seemed to be related to the activity level of the swarm or number of mosquitoes present in the swarm. When it comes to host odor, it seems to have an effect on swarm activity and duration. So what do we need to do now? Well, everything. Because, one thing keeps opening up more questions with this project, that's for sure. But in the near future, I want to figure out what this lactone 5 has, if it has a specific role, or maybe it just doesn't, maybe it's just there and it doesn't have a specific role. Either way, I'm gonna look into it. Then the other thing is that we want to look at the threshold as well. So we want to do odor collection with different amounts of males to see if the profile changes. Could it be that when you're reaching a certain point, they're releasing other odors, etc. So we want to take a closer look at the whole old-fashioned threshold of male grouping. Of course, we want to look at the role of human host, because even though I've been saying that host cues have this effect, there are other things that could have this effect, such as temperature, humidity, of course. So one of the things that first we need to do is to perhaps introduce host odor without the actual host. This way, we eliminate temperature and we eliminate humidity. Okay? Once we have this, then of course the aim is to know the molecular work. So we would like to find out the receptors, etc. But yeah, maybe that's for the next meeting. And with that, I would like to thank Hong Lei, who helps me with the chemistry, the GCMS analysis. Of course, Marcus, for being Marcus, and Formas, our funding body. And thank you guys for listening.
Thank you, Nadia.
Yeah, so under the everything category, are you gonna start looking at combinations of lectins, like lectin 5 and 1?
Yeah, so we would like to quantify first after each scenario. And once we have that quantification done, it would be fun to play around with it, of course. Yeah, would be nice.
Yeah, thank you for your talk. First question, do you guess which is the source of these lactones in the mosquito? Where do they produce it? And then the second question, have you tried, instead of ham, host feed fodder? Because at least in my experience in Brazil, they offer you feed first. So, I mean, thinking on how much can you get in terms of synergistic effects?
Yeah, of course. I mean, and for the first question, we want to do labeling and analyze that in the GCMS. So then we would know the whole biochemical pathway of how that would occur. So that's the aim. And host, yeah, I mean, that would probably be the way that we try to do host order without having the hand. So a nylon sock, the old school way, yeah.
Thank you.
Thank you for the talk. It was beautifully put together and illustrated, and I enjoyed it. The one thing that I noticed was when you have the hand in there is a very distinct visual stimulus. And for most insects, both blood feeding and plant feeding, there's a sort of building the shaft of when you have more.
Yeah.
So, are you going to sort of stick an artificial mannequin hand in there without the odors? Because we stick anything in a mosquito cage and it will disturb them and get the fly.
Of course, definitely. I mean, vision has to play a role as well. So that's something that we have to do, I think, in order before we can determine anything. We have to take into consideration vision. So yeah.
We have a question from
online, yeah. From Marta, with saying thanks for the nice talk. And I would like to ask, how would you define swarming in AE Egyptian mosquitoes? Do you see circadian time dependent differences?
Yeah. So definition of swarming, I guess this is something that we were talking about yesterday. How do we define swarming? Well, at the moment in the lab, because it's a smaller cage, that they're active. So that's our definition at the moment. That they're active and they're flying in a pattern. That's what we consider. Of course, in nature, this probably differs. They're much bigger. So it's just, at the moment, that's our definition about swarming. Yeah. And in regards to the other one,
Circadian time.
yeah. So we've noticed that males are definitely more active on said photophase. So basically in the lab when the lights go on. So all the experiments are carried out during the first hours of the day. So once they come down a bit, then we start the experiments. Otherwise, they don't do very much later on. So yeah, that's what we can see.
Yeah, thank you. I have a question for the audience. Who has really seen swarms in the wild?
Yeah.
With Aedes aegypti or Aedes albopicus. I spent 30 years with Aedes mosquitoes and I got several viruses. I never seen. So who has seen? And who is having? You are the only one. Can you explain what you have seen?
Yes, that was in Burkina Faso many years ago. And and they were forming at the base of the tree in a compound.
Okay, not around here.
Gentlemen, and then Daniel.
Here you go.
Yeah, interesting. Have you considered introducing a group of females? Because females tend to change the behavior of males.
Yeah. So before we did this, I did a lot of, in another kind of cages that we have, where I had females, and then males had the possibility to get to the females through a tunnel. That didn't happen. At least it wasn't statistically significant. I did the other way around as well, where females were able to go to the males. We didn't see much of results there.
In terms of measuring activity, like you did with the hams, but then.
No, then we were looking at attraction kind of thing, but I have not seen, we haven't put females within the closed environment. Yeah, no, we haven't done that yet.
SPME data is very difficult to quantify.
Yeah.
Methodology.
Yeah.
How much do you think these insects produce, and is what you're testing in the bioassay similar to the amounts that insects produce?
Right. So as you said, it's difficult to quantify. What we can quantify is the amount collected in the conditions that we had. And we did that. And at the moment, we are testing a higher concentration than what would be equivalent of what supposedly is collected from what we've done. So, yeah.
So like 10 times or 100 times? Or...
Yeah, it's probably like 5 times more concentration.
Last question for Carol.
Yes. How do you know that these compounds are actually attractive? What do you define attractiveness of your testing?
So as I showed in the Uniport, attracted for us, and this is an assay that we've used for all of our behavioral studies in the past for females as well. In that little chamber there that you see attracted, there's a cone, right? So the mosquito has to actually make its way through the cone, which is like a little trap, in order to get there. Once it does that, then we consider it attractive. So we consider it attractive once they made the effort to go the entire way there and reach the owner source.
Would any increase in the linear velocity of your mosquitoes increase the likelihood of being attracted in your bioassay, much as the same way they could go back into the unactivated chamber? There you go.
I don't know. I'm not quite sure I understand. Like if you would change.
Well, maybe they're just increasing the linear speed of these mosquitoes. And so by chance, they get into the attracted chamber in much the same way they could, because they're moving more and their speed, they're faster.
Yeah.
They could go even back into the unactivated chamber.
Yeah, so then I guess if we're
So
thinking
it would
about
not be an attraction by itself. If this interpretation is correct, you may think that something that gets these males away from other males.
Right, okay. Well, I'm not sure. I mean, the way I would say in regards to that is if it's by chance, that's why we do a lot of repetitions to try to confirm that it's actually happening. I don't know if that answers your question, but yeah.
Or you can just remove the tray.
If I just remove the what?
You can remove the tray and see if they stay in the area.
Or simply measure the linear speed. In your assistant.
Yeah.
On that point, just maybe a dual choice, an empty box.
Yeah.
And then, right.
Like a white you kind of
Yeah.
scenario. Yeah, definitely. Yeah. That would be a good way of testing.
Really last one for me.
Quick one, Philip, do male and female human hens have the same order profile?
I don't know. I'll have to ask Marcus to put his hand in it. So far it's only mine.
Thank you very much, Nadia.
Thank you.
So my talk could be about mosquito acoustic communication. and its implication for sound traps or its potential implication for sound traps and mosquito control. The sound can propagate into different mediums, such as water, so these sounds could be detected by larvae, but I'm not going to talk about this. Or sound can also propagate into the air. So adults can detect the air, the airborne acoustics. Hearing in mosquitoes is involved in different behavior, such as mating during swarming behavior. In most of the species, at least in males, in the search of blood hosts in some species, and maybe in predator escape. So in this presentation, I'm going to see how we can manipulate this behavior to make sound traps, or more generally, how to potentially control mosquito with sound. So I will first present how the hearing mechanisms work, then what organs are sensitive to, so in terms of electrophysiology, then behaviorally what mosquitoes respond to, and finally the implication for sound traps. So this is the head of male Anopheles mosquitoes. So you can see the two flagellum with the fibrilla, the hairs. So this is a male. A female is basically the same, except that the hairs, the fibrillae are far less and far smaller than in the male, but it's basically the same. They are the same organs. So the mosquitoes can detect the particle velocity of the sound from the hairs here, which are coupled with friction in the air. The vibration propagates to the flagellum. So this flagellum has a quite broad frequency response. So here you can see in males, so in blue, and in red for females. So more or less from 200 Hertz to 1 kilohertz, more or less. So quite broad range. So this vibration then goes to the basis of the antenna, and so this is a Johnson organ, kind of donut, where the antenna can deviate around, and the Johnson organ transforms the mechanical vibration of the antenna into an electrical signal. So the Johnson organ has a far narrower tuning compared as to the antenna. So here this is a little bit threshold. So the smaller the value, the better the hear. So this is the Johnson organ literary threshold for male here. So what you can see that it's very narrower than the antenna frequency response, only a couple of hertz here. So if a male is resting, and if the female wind frequency is diffused into the air, you can see that a male cannot hear a female very well because it doesn't fit with the tuning of the Johnson organ. However, if the male starts to beat his own wing, then there will be a non-linear vibration inside the antenna, and there will be a distortion product which will be created in the antenna. So in the antenna, you won't have only the female wind frequency and the male wind frequency. You will also have a quadratic distortion, which is equal to the difference tone, the difference frequency between the male and the female wind frequency. And the Johnson organ is tuned to this difference tone. So it's a little if you would like to hear my voice, you will have to speak at the same time in order to hear my voice, and to adjust your pitch, your voice pitch, in order that the difference frequency between your voice and my voice is more or less 300 Hz. If you do that, you will manage to hear me better, I think. So this system is highly sensitive. So it's reported to be at least one of the most sensitive auditory organ among all arthropods. So besides, in addition to the distortion product, which boosts the hearing of at least males, so they use their own wing beak frequency to amplify the sound of the nearby mosquito. So in addition to this, they also have some self-oscillation of the antenna, at least for males, at the female wing beak frequency, so that they can pick up very tiny, very quiet sound in the air. So most of the studies have been done on males, but we expect that it's similar in females as well. So in terms of frequency, so about the organ, so I gave you this example with sound frequency of female and male. So males can detect the females through the distortion product, but they are not the only frequency that mosquitoes can hear. So for instance, if we have two sounds, so X and Y, mosquito will be able to hear this sound, provided that the difference frequency between X and Y is equal to 300 Hz approximately. So from the distortion product, the mosquito will be able to hear higher pitch through the distortion product. So this is right only if the higher pitch, so the X and Y, are within the frequency range of the antenna. So they should be less than at least 2 kHz. So for instance, mosquitoes cannot, with this hearing mechanisms, they cannot hear ultrasounds, for instance, because the antenna doesn't vibrate to this frequency. So mosquito cannot hear ultrasounds, at least with the adjacent organ and antenna. And second, providing on enough sound level. So there is a trade-off between the power of the sound source and the distance to the sound source. So they can hear very quiet sound at short distance, like a single mosquito, and they may hear more powerful sound at longer range. So theoretically, from the electrophysiology, for instance, they could hear a melting swarm because this is in the range of their frequency sensitivity. So if the sound is powerful. So now, in practice, behaviorally, what do they respond to? So first, so the something which is known for a very long time, that male move toward the sound source of the female. So if you play back the female frequency, the male will move toward this frequency. So that can happen with the speakers, but also with real mosquitoes. So with very quiet sound. So this is used in the swarm. So when you have male swarm, when a female joins the swarm, the male will detect the female from the sound, and then there will be a chase which will start. The female will lead the chase, and the male, at least, are expected to detect to localize the female just from the sound. So this is an example of 2 mosquitoes swarming in the lab. Here I think you have the male, the female. So they are swarming above, so they are Anopheles mosquito, quality. No, Culex, I think they are Culex, they are swarming above a swarm marker. So at the beginning, they are not supposed to have detected each other. And when they get close enough to each other, they start this chase. So yes, the pair was so they didn't manage to copulate, but there was a pair at one point, and then they start again the chase. So we have just shown with so Olivier Roux and Gabriel Gibson that behaviorally this hearing threshold can be quantified, and it's less than 20 decibel of some particle velocity level, which is very quiet sound. So with the similar hearing mechanisms that I talked just before, we can expect that male can detect each other in the swarm in order not to crash into each other. So this has not been really shown, but one study suggests this. So if male change their radio frequency in order that there is a frequency difference between each other, they may hear each other. But that's been shown. You may have heard about male-female harmonic convergence. So this is defined as a way for male and female to test their fitness from so just by moving their winging frequency, and it's believed that this model says that when they're convergent, it means that they think that the other is has a good fitness. So this has been shown on Asian mosquitoes. I don't think it has been measured on free-flying one. But this theory has been challenged. So first, one study, Simwise and colleagues, suggests that this could be just an EP phenomena of the hearing mechanisms. So mosquitoes just try to shift the frequency in order to hear better, and by chance, this falls on harmonic convergence. So this is one alternative explanation to harmonic convergence. And the second one, the more recent studies by Summers and colleagues, which so they suggest that this could be not related to any interaction between the two mosquitoes. So harmonic convergence can be measured. If you have one male, one female in different rooms, you can measure it. But there is no interaction at all. It's only controlled by circadian rhythm. So male would shift their winging frequency at the sunset time, and if you measure the different tone, it's you have a ratio of 3:2. So you can call that harmonic convergence. But they show that there is no interaction between the two mosquitoes. Other behavior, like when you play back a sound from a speaker, sometimes when males are swarming, they just stop moving or change their angular velocity suddenly. So this can be explained by the fact that the wave front is plane, as compared to a mosquito, the real mosquitoes, for which the wave front is spherical. So then the mosquitoes, when he encounters the wave front, he cannot localize the female. And then he just doesn't know where the female can hear it. Finally, so still with low power sound. So male rapid frequency modulation has been measured many times in many studies. So this is something which seems to happen just before or at the contact between male and female. So we don't know yet why it happens. Some hypothesis is about in order to stabilize the couple in flight, because they are still flying when they are in pair. So that would be maybe a way to stabilize in terms of more powerful sound. So one hypothesis that we raised was that female in electrophysiology, they are sensitive to the male sound. They don't react to a single male sound at all. There is no movement toward that. So the question was, maybe they could react to the male swarm sound, which is a little different, broader frequency range. So we tested that, and we showed that at natural sound level, at least in NFL quality, females doesn't respond at all to natural, swarm sound. It has been shown that, particularly in AD species, that male and female are repelled by low frequencies. So in males, here by Lapshin and Bronsoft, And in second study by Duet et al., it has been shown in female, but it was with very loud sound, like 100 decibel. So yes, this has to be shown again with the field with predators. And finally, in a couple of species, females shows phonotaxis behavior too, so they are attracted to the sun source. So usually, sometimes there are frog beating mosquitoes. So this has been shown in several studies. So now, what are the implications for sun traps and mosquito control? So first, we can catch. So could we catch mosquitoes with sun traps? So if we look at this diagram, so there are frequency range of females for about 20 species, and you can see that depending on the species, you may split the different species according to their winning frequencies. So at a given location, if you have the correct species, you may use a sound trap with a particular frequency, which will be species specific. But of course, it depends on the location. If you have these two species together, you won't be able to, your traps won't be species specific. So sound traps, so it's not something new. So the basic technique has not changed since the first reported traps, so by Maxim in 1901. You just play back the sound frequency of the female, and you catch the mosquitoes with a fan or with some glue. So it has been shown to be efficient on many species, male or females. So there are some reports that say that it's very, very efficient, but sometimes, yeah, it's difficult to get an opinion on this, because when you have the sound traps, you also have the speakers, and you also have some visual cues. So mosquitoes are also attracted to visual cues. So it has been also studied the influence of the two, or the synergy of the two. So it has been shown to be, in one study, as good as a standard CO2 traps in Aedes aegypti males, and not as good, but almost as good in another study, but only in Aedes aegypti males. So as CO2 traps, they are not, they cannot significantly reduce the population, at least beyond the restricted area. If you use multi-modality traps, so if you add to the sound and the visual cues CO2 animals, you can obtain some nice results and some synergetic effect. So this was catching. So now could we repel mosquitoes? So it has not been tried. There are just one study, maybe a couple of studies. So the main issue of that is that the mosquitoes are sensitive to sound that are also sensitive. And if you want a long-range attractant, you will need a very loud sound. So in practice, it's not very nice for people or animals overall. Killing. So with sound, you cannot kill the mosquitoes except if you play back really very, very loud sound, but then it will destroy also other things. But maybe what you could do is maybe fatigue mosquitoes, maybe. So it will be more about how to disrupt, for instance, the mating swarm. But you are not going to put speakers on each swarm that you see, so it's impossible. So you will have to think about traps with maybe a swarm, hyper-stimulus swarming marker, where in order to attract more mosquitoes, and then you can use the sound. But it has not been done yet. Just like that. So to conclude, some traps in practice are only short range. They work well with other modalities, but they work well with males of most species. For a few species, it works with females, plus or minus species specific according to the given location, the given species that you have. There is some synergetic effect with the swarming marker and other cues. And so, for an example, it could be used to monitor the survival rate of release sterile or genetically modified, and it works well for future research. So that could be about looking at mating disruption with the sound and hyper swarming stimuli. And there is something that I find quite interesting in the future is there is a female hearing paradox. So most of the females are very sensitive to sound in electrophysiology, but they don't show any behavioral change to sound, no response at all. So why is it that? Have we missed something? So this should be if you want to know more about acoustic communication mosquitoes, we have just written this review paper with Patricia Simoes, Ian Russell, and Gabriel Gibson, and it will be released soon. That I can send you author version if you like. Thank you very much.
Thank you. Do you have any questions?
A very general question. Thank you for the presentation. The sense of what's the range of their condition capabilities, how far away can you?
So it really depends. So there is no limitation of the range.
Theoretical or in practice?
No, in practice, so they are sensitive to particle velocity, which is a component of sound. So, so the question is more about loudness of the sound. If you play something very loud, they will be able to hear it at 10 kilometers. If you play something,
so mosquito language.
Yeah.
So in terms of the power that mosquito, the output of the mosquito wing beat, that's the range which they can communicate with the mosquito.
Yeah.
I don't know anything.
Yeah. So in terms of mosquito sound, I don't think they can hear at no range. So if you take a single mosquito, it's very quiet, so that would be a couple of centimeters maybe. If you take a swarm, so this is what we try. So we try to study the loudest swarm, so in another quality. And the male are not expected to hear the sound, but the female could use this sound to detect the swarm, and they have not shown any behavioral response in terms of wing frequency change, linear or angular speed.
So we could go from 2 centimeters to much longer distance with the swarm. Is that what you're trying?
No, for the swarm, we were testing, sorry, at long range. If the female were able to hear the swarm sound, because the swarm sound is quite loud, far louder than a single mosquito. So they could hear it at long range, because the electrophysiology say that they are sensitive to this frequency, but they are not responding. They have no range.
That long range would be, I'm sorry to push that, that long range would be what, 20 centimeters?
So we tested, we tested more like up to 1 meter. Thank you. So it was with playback, but it was equivalent to 1 meter.
That has to be very sensitive.
Yeah, thanks for the talk. I was wondering, you said that females can hear this, they can hear, but they don't respond. Do you know if anyone has tried to test female host-related behavior with the background of male sound, whether it could have an effect on this under these conditions? Because for the female, it might make sense to have a host in the close range.
So in order to get this discussion,
changes in female behavior.
I'm not aware about this. So that could be as a case for ADs, where the female blood feed nearby the male, but
Any questions on sound? Do you think that the sound effect could be dependent to the age of the mosquito? And especially, is it possible that some traps could attract differently young versus old mosquitoes?
So I think there is some small effect the first day. So there is maybe some change between as the mosquito gets in adults up to the 5 days old. I'm not sure exactly the number. There is some small changes in how they are sensitive to sound. Also, their window frequency change a little when the first day. but otherwise I don't think it will change phenomenally.
Do you know of any sound that we produce, voluntarily or not, that they will be sensitive to?
Yeah, so you mean humans?
Yes.
Yeah, so there was a study about long-range hearing in 80s, and so they play back female sound at very long range, and they show that the male were responding to that, and then they concluded that they can hear at long range. They couldn't hear this mosquito sound because it's too far, it was artificially loud, but they are the same frequency as the voice. So the windy frequency is more or less the same as our frequency range, or voice frequency range. So yeah. I think they are sensitive to the fundamental frequency voice. And actually, yes, sorry, when you are next to a swarm, male swarm, at least in QX, Pachatis and fertility, if you sing the female pitch, it's very clear the male mosquitoes will go to your face. So that's because they are just the same frequencies.
Yeah, relevant to the question about what sound is doing to the females. So we knocked out a trip channel that renders the Aedes aegypti deaf, both males and females, and the female mating is very delayed if they're deaf. So hearing must contribute to their mating behavior.
Yeah, so that's the in cages.
Well, yeah.
Yeah, so in cages, if a female is deaf, so she can fly, the male can detect the female.
Yeah, their locomotor activity is normal, not just based on their trikinetics, but also using video recording. So their activity is normal.
My guess is that female could use the sound of the male during the mating checks, but if she deaf, I don't know, because she could use the sound in order to escape the male if she wants to. So that's not in the same direction as your idea.
I think it would be nice to test this strain with your devices of recording themselves. Perhaps if they need to hear themselves to emit the sound, perhaps if they are deaf, they don't hear. So
their fundamental wing beat frequency is unchanged if they're deaf. And also, if you have one deaf partner, they'll still harmonize. The other will compensate, and you'll get normal harmonic interactions. Even one is deaf. The other won't change their frequencies at all, but the other that can hear will make a full adjustment to harmonize.
Male or female?
Male or female, goes both ways. You only need one hearing animal for harmonization.
Yeah. So I get that, yes, Somers and their colleagues will say that maybe if you get so the female or the male deaf, maybe it will change the circadian rhythm, and then maybe they will harmonize less because they will not increase as much as they will not increase their frequency as much as mosquitoes, which would be in the correct circadian phase.
Yeah, it doesn't seem to be related to circadian rhythm at all. So we can reproduce what Hoy published in 2009, and but obviously if they're both deaf, they don't harmonize at all. But you only need one to get the harmonization.
Last question from speaker line.
Yeah, from Martha. Do you think that acoustic traps could be used to attract mosquitoes in the long range?
So they could, but it will require a very loud sound. So in practice, I don't think it's could do it. But I think even yes, the earliest traps from Cannes in 1949, they reported that their traps were more efficient if the sound was louder, and they also reported that it was very irritating for everybody during the experiment. You need something very powerful.
perhaps we could implement this in festivals, but there is a lot of all the mosquitoes. Thank you very much.
They told me I have to speak, so here I am. As David said, I'm going to tell you a story about the effect of knocking down IR8A, which is a co-receptor for the ionotropic receptors in insects, that has an impact on the expression of robust sexual behavior in males of Brownies perixus. Well, Romina did a nice job yesterday introducing Brownies perixus, so I will not spend much time on this. It is a very important vector of Trypanosoma cruzi, the causative agent of Chagas disease. But it has been also a classic model for insect physiology studies since almost 100 years ago, because it was the model with which Vincent Wigglesworth described the endocrine control of molting insects. So these are hematophagous insects, mainly hematophagous. We know that they can bite other arthropods and get a hemolymph from them, and they are nocturnal. They spend most of their life hidden in cracks in very narrow shelters. They don't show up unless they detect relevant cues or signals, depending whether we're talking about starved insects that need to detect the host in order to leave their shelters and engage in foraging, or adult insects, for which, in the case of males, we have shown that they detect the presence of females and react to that by leaving their hiding places. So these are hemimetabolous insects with 5 instars, the larval instars, and then the adult stage. They all need to engorge on blood in order to molt, and the adults need to eat frequently in order to produce eggs in the case of females, but also in order to be properly acceptable for the females in the case of males. And they use the olfactory system for recognizing hosts, of course, even though they are multimodal, and they use heat and humidity also as cues of the presence of the host. These are very important cues for triatomins. But in the case of adults, they emit alarm signals in case they are disturbed manually, and they use sexual pheromones. We have shown that a while before in order to promote mating encounters. So the metasternal glands of Brownies perixus are a source of a complex blend of compounds that we have shown that act as a sexual pheromone. In the case of Brownies, they are fundamentally a blend of alcohols with traces of ketones, and there is no difference between male and female secretions, which is quite curious because only virgin females emit these compounds during the first half of the dark cycle, of the dark phase of the daily cycle, sorry, and they emit the compounds when they are fed, so they are motivated to mate. They also emit these compounds during mating, and we know that during mating it is important for both females and males to have intact metasternal glands. So the metasternal glands, you can see a schematic representation here. This is the thorax of an adult Brownies perixus in a ventral view. So you see there is a tubule and then a reservoir. They are parent glands, so there is another gland on the right side. And these glands produce this blend that promotes the activation of shelter mates, as I mentioned. So when males detect the presence of female odor in the environment, they actively leave their shelters, they engage in female search, and they show odor modulated anemotaxis in response to female odors. We have been able to show that the males present a significant increase in takeoff responses when they detect the presence of female emitted odors in air currents. So this is a parenthesis. Domiciliated triatomins have been said for a long time that to be bad flyers or non-flyers at all. But we have shown in this work that indeed Rhodnis prolixus flies quite frequently, up to 30 or 40% of the adults, both males and females, tend to fly in a single night if you offer proper conditions. But in the case of males, this frequency of takeoff more than duplicates in the presence of metasternal gland compounds. As I said, this secretion is important for mating, for observing proper mating in this insect. So if you block the glands of either females or males, you see a decrease in mating frequency. And then for 50 years, we knew that there was male aggregation promoted by mating pairs. So when you have a mating pair, other males tend to aggregate, like in this picture. You see, this is a mating pair, and these other males are visiting the area and stay close to the mating pair. And this has been claimed to be a form of polyandry, at least for Triatoma infestans, Triatoma brasiliensis, and Rhodnis prolixus. There are other triatomins that do not show this kind of behavior. But these insects can mate frequently. And in the case of male aggregations, we have observed repeatedly that the female can mate with more than one male in a sequence. So this is more or less general perspective on the sexual communication by means of sexual pheromones in Rhodnis prolixus. But when we started this work, we didn't know anything about the molecular basis of pheromone detection. We still don't know much, I should highlight before telling you the story. But we now started to disentangle what we think is behind this detection. We were part of the team that described the genome of Rhodnis prolixus, and together with Professor Hugh Robertson, we were able to describe a set of genes that are either related to sensory processes or to behavior. And among these genes, we were able to detect the ORs and IRs of Rhodnis. And a bit later, we published a paper showing the expression profiles of all these genes in the antenna of 5th instar larvae, so the last larval instar, male and female insects. And you can see here examples of what we have seen from low expression to high expression in yellow. You can see that ORCO, the co-receptor of the odorant receptors, is much more highly expressed in the antenna of female and male insects compared to larvae. We have seen a similar profile with other odorant receptors, which we can, propose that are potentially related to some adult related olfaction process and probably related to the detection of sexual signals. But we have also seen that two ionotropic receptor co-receptors, IR8A and IR76P, are quite more highly expressed in the antenna of males than females. And this was at least curious for us, because they are ionotropic receptors are normally not claimed to be related to the detection of pheromones. But we decided that we would give it a try and try to knock down the expression of IR8A. IR8A is expressed mostly in neurons housed in coeloconics and cilia, and these neurons and these ionotropic receptors have been related to diverse sensory processes, olfactory, gustatory, even thermal, and detection processes like you heard yesterday. And we know that in many cases, expression knockdown or knockout will promote behavioral phenotypes. And that was what we were looking for in our experiments. So with this in mind, we wanted to test whether IR8A had a role in the sexual communication of Rhodnis prolixus, and specifically, we wanted to see whether RNA interference would promote an effect on male trigger activation, male activation. So whether males that have IR8A knocked down would have lower tendency to leave their shelters and engage in the search for females, and whether mating frequencies will be affected in the case of knockdown males. For this, we used males and females that were sex sorted during the last larval instar. So they became adults and they were kept virgin until the behavior experiments. In the case of males, we injected the sRNA to knock down the expression of IR8A in males that had between 1 and 4 days of age after ecdysis. Then we fed them around 10 days later, and we tested them between 5 and 10 days after feeding. These are conditions that we know grant a highest motivation for mating. And in the case of females, we just fed them because this is critical for females to accept mating, and then we tested them at the same age as males. For knocking down the expression of IR8A, we injected the sRNA intraperitoneally by means of a Hamilton syringe. This is a method that has been used widely with triatomines, and triatomines are a very nice model for RNA interference. It works quite nicely. So we applied 1 microgram of the sRNA targeting IR8A. And we performed experiments with 3 groups of males. Males that were not injected, so intact males, males that had an unspecific dsRNA designed not to knock down the expression of any insect genes, ampicillin in this case, and then dsRNA for specific for targeting IR8A. And our knockdown on this receptor in the antenna is very effective. We have more than 80% decrease in expression of this gene. And we first used this approach to test whether a male behavior would be more or less normal in injected males. So we used the proboscis extension reflex, which triatomines show as a response to the presence of a heat source that resembles the temperature of host. So this is normally expressed in intact insects. We wanted to test whether the injected insects would express it similarly. And we did this for all the males used in the experiments. We saw a similar percentage of proboscis extension reflex in all 3 groups. So there seemed not to be any other behavioral effect, but we wanted to test that only also in whether feeding was affected and males fed normally. The amount of blood ingested was similar to that of intact males. So for the first hypothesis, we used a very small arena with an even smaller shelter and an empty flask glued on the side of the arena, and we video filmed the males 3 days after introducing them into this arena. So to make sure that they were used to the environment, that they were able to exploit the shelters, and under such circumstances, we measured in the first night. I should say in the 3rd night, we measured the percentage of males outside the shelter and the percentage of males that stood on the mesh of the flask. This empty flask was used in the following night to introduce a female in the experiment. The female was introduced right before lights off and left for the whole night, and the behavior of males recorded for that night also. And we compared what we saw in both conditions for the 3 groups of males. And what we saw in the first night with no female odor present is that what we expected normally, males, around 10% of the present males would leave their shelters and not more than that. But when we introduced the females in the following night, what we observed is that the 2 control groups of males recognized that there was a female in the area, and they reacted by leaving their shelters and engaging in search, but the knockdown males failed to do so. And when we looked at the percentage of males standing on the mesh of the flask, so where the female was, we saw a similar result. So this is the percentage of males that left the shelter and that managed to find the female. So according to these results, we suggest that IR8A is necessary for activation for the activation of males that need to leave their shelters, and also for the finding of the female. And in a second experiment, we tested whether the mating frequency of pairs, including knockdown males, would be affected through 40 minutes of experiments. And we used 40 minutes because the mating of these insects takes quite long, up to an hour. So you can see that in both control groups of males, of pairs, including control males, there is a similar dynamics with most of the matings starting in the first 10 minutes. There is only a very slow accumulation of mating pairs being added to these numbers after 10 minutes. But when we tested knockdown males, not pairs that had knockdown males, the result was quite different. So, especially during the first 10 minutes, we failed to observe this drastic increase in number of mating pairs in the pairs that had knockdown males. So, in our impression, and this shows it in a different way, this is the percentage of mating pairs in 1, 5, and 10 minutes after the initiation of result of the assays. You can tell that the pairs including knockdown males failed to show normal, proper mating behavior. And I should highlight that in geodomins, and particularly in Rhodnys prolixus, it is only males that attempt mating and that initiate the mating interaction. So it seems that knockdown males, IR8A knockdown males, are much less capable of recognizing that there is a female in the small arena where they were tested. So to conclude, I would suggest that IR8A is necessary for proper chemical communication in the mating context of Rhodnys prolixus, that male activation is lost in the presence of female others after IR8A has been knocked down in males, that these males fail to find the females in this simple experiment that we performed, and that mating frequency is dramatically affected by by impeding the expression of IR8A. Our proposal is that there seems to be a lack of pheromone trigger arousal in knockdown males, which is how we interpret what happened in this specific, especially in the second experiment that I showed you. I would like to conclude thanking Jose Latorre, who prepared the dsRNA for these experiments and did the qPCR to test the knockdown, whether knockdown would work. Alessandro Guarneri, who did the DS injections on the adult males, and then Gina Pontes, who together with me performed the behavior experiments in Brazil. Of course, the funding sources that helped me perform these experiments, and I would like to also thank MAK'IT for making it possible to be here to sharing these results with you. And I would be happy to take questions if you have any. Thank you. Yeah.
Beautiful, Marcelo, thank you. Do you think that, or maybe did you test the response to other odors related to a different behavioral context in order to show that it is not the whole olfactory system shut down and it is specific for male or mating behavior?
Thank you for the question. It is super relevant. We don't claim this is specific for sexual behavior or for sexual signals, because this is a co-receptor. So any specific receptors co-expressed with it will be affected. And of course, we don't think, as I said, Rhodnys prolixus has 33 ionotropic receptors, half of which are specific lineage, which is mostly duplications of IR75A. So I would say probably host-related other detection will be affected, even though they feed properly. So that doesn't seem to be critical. But yes, I would not claim that this is the only effect. One important thing I didn't show you is that there are many IRs that are more highly expressed in the antenna of males. So there might be more than one specific IR related to this effect.
That was my question. I have a more important question.
Yeah.
When I do the manual injection of CRISPR gRNA in the eggs of the rodents, because this is so big, how big are the eggs?
The eggs are rather big. So far that I know, nobody managed to do CRISPR with germans. We would love to do it, but the eggs are quite hard and yeah, I think most people fail. I know people were but it's quite difficult to do. So so far I don't know of anyone having success with CRISPR, unfortunately.
What kinds of anatomical sexual dimorphisms are there in the pubertal active organs, and also what kinds of 8A associated co-receptors are present in body?
I'm not sure I heard your question. I mean, if you mean dimorphism, we
Anatomical dimorphisms are there specific types of cincilla that are present
Okay.
in the males versus the females, and also whether the IR8A partners that are known in flies and other insects, which of those are present in bodies?
Yeah, all right. We don't know much about sensilla related to sexual behavior in these insects. In part, this is due to the fact that they have many neurons per sensilla. So doing electrophysiology, I don't want to use the expression, it's super difficult. Most people have failed. We have failed. We have tried to do electrophysiology with the sexual pheromones. We get kind of responses, but it's very difficult to tell who's responding in that sensilla. So no, we don't know if there is dimorphism in terms of types of sensilla that are present in the antenna. We do know that some morphological types seem to be to present different proportions in but it's very weak evidence.
I was wondering if, so you showed that by suppressing genes, there's less mating. I was wondering if you went on and analyzing those that did mate, if the mating was successful and the females were actually inseminated. I'm just wondering whether it's suppressing the initial phase of the behavior or if it's a knockout of the whole behavior.
Well, we did, and we tried to keep these insects. I mean, the offspring. We're trying to keep them because for some, at least for some genes in tree elements, it has been shown that knockdown by means of RNA interference is preserved through generations. So we want to test these insects to see whether they are still showing this lack of expression, but we don't have any results yet. But yes, they can mate. I mean, a very low number of them mate, and apparently the offspring are from the eggs. Yeah.
So you mentioned that the females are polyandrous. Do you know if the progeny could be from more than one father?
That's a good question. And nobody knows. I mean, nobody has done that. So it is super important. We have been thinking that we should pay attention to that, but we don't have any results yet. Yeah, it's super relevant.
Okay, thank you very much, Marcelo.
Welcome back. I
I hope you had a good lunch. French cuisine is always on spot. So this afternoon we will start with the session on repellents for the behavioral manipulation of vectors. And our first speaker is Nicoletta Faraone from Acadia University. Hello.
There you go. Okay, welcome everybody. Thank you very much for the invitation, Marcelo and David. So this will be the first and the last talk about ticks, I think, because there are no other talks about ticks. So before to start, first it's not working now.
Fix it.
Yes. Okay, before to start, I would like to show you where Acadia University is located. So we are in Nova Scotia, so in the east coast of Canada. We are located at the top here in the so-called Bayou Funding, where we have the highest tide. There is a 16 meters difference. It's very nice. It's a small town, Wolfville, with lots of wineries, so come to visit. It's a lovely place. So then, for not forget, I want to thank, first of all my labs, all my students and postdocs and collaborators that are make things possible, because are actually the people working in the lab and doing the hard work. And of course, all the funding agencies that are supporting my research. So I would like to start showing you a beautiful picture of an Ixodes scapularis nip. So you can see here, it's a close-up of the mouth part with all the sear. This is all the shape, the sears that is basically meant to anchor very strongly on your skin or the skin, the host skin, for the feeding process. So why it's important to study ticks and why it's important to study the chemical ecology of ticks. So we're all experiencing the effect of climate change, unfortunately, and the overall global warming. And this is going to get, it has promoted the emergence of a vector-borne disease. And one of the most serious vector-borne diseases is so-called the Lyme disease, which is caused by a bacteria called the Borrelia burgdorferi that is transmitted in North America by Ixodes scapularis, or black-leg tick. Here you can see that also in the Northern Hemisphere we have lots of cases of Lyme disease, and in Europe and the other part of the other side of the pond, the species is the Ixodes ricinus, that is very, very similar in morphology to the Ixodes scapularis. Now, the Lyme disease cases have been increasing exponentially in the past 10 years, and you can see that now we are having almost 2,700 cases in Canada, and this is getting the public health attention because, of course, it's a disease that is difficult to detect, and if it's not properly treated by a course of antibiotic, can lead to permanent injuries and can be life-threatening. Now, Nova Scotia, where I live and there is Acadia University, is considered one of the, located there, it's one of the highest, it's one of the provinces in Canada where has the highest risk of contracting Lyme disease from a tick bite. And you can see here these two maps. They are not updated in 2022 because, of course, it was the pandemic in the beginning, so everything stopped. But you can see here that the counties that are colored in dark blue are those where there is the highest risk of contracting Lyme disease by tick bites. So the situation in Nova Scotia is very, very serious. So that's why we need to try to solve this tick problem. So the risk of contracting disease from a tick bite has led to the search to find effective and environmentally friendly tick repellent product that will protect us against a tick bite. So I'm very much more on the prevention side here. And natural products and essential oils offer a good alternative to the commonly known as synthetic pesticide. But to find the right active ingredient, the right product that will be effective in repelling ticks implies that we know well how ticks perceive repellent, how they process the signal. So we need to know very well how tick chemiosensory system works. So how does it work? So until recently, very little work has been done trying to understand the tick chemiosensory system. So what we know is that tick use mainly the very unique organs that is found in acarines and specific in ticks called hallmark organs that is located on the front tarsi. It's mainly responsible to detect volatiles. And only recently we have documented the involvement of pulps, pedipalps, and the mouth part in also detecting close range volatiles. Now, it's not really clear how this process works, attractant and repellent detection, if it's only olfaction or also contact is involved, and the mode of action of this volatile organic compound, how they are processed, because ticks don't have ORs. So the detection is mainly done through IRs, and there are some GRs. So also the repellent action is a true repellent, or there is a deterrent in addition. So there is a lot of question that needs to be answered when we look at the tick system. And another question that for me is quite interesting, I don't know what this should be a tick, but it's a mosquitoes. My presentation would be, cryptic with mosquitoes. So think about there is a tick there. So another important, this is funny. Another important question that I would like to answer is how the microbial infection has an impact in the tick response to volatiles. Because the majority of the work out there has been done on ticks that are lab-reared, so they're infection-free. So we know that particularly Ixodes scapularis, that is infected with the bacteria Borrelia burgdorferi, is way more resilient to fluctuation in temperature, changing the humidity level. Ticks really like humid environment, so dry environment may affect them, but those that are infected are stronger. They quest and they seek for hosts in a more aggressive way. So how this impact the way how repellent may work, right? They can be effective. So based on this interesting question, my lab focused mostly on trying to answer, to understand what are the compounds, the volatile organic compounds that are behaviorally relevant, and looking at host odors and also repellent compound, focusing on natural products, and trying to study this from a neurophysiology level and also through a behavioral level. And with this information, we want to develop a new repellent and acricidal product, exploring nanoformulations or nanotechnology to maximize the activity of this product. So for this presentation, I will mostly talk about this and leave the chemistry for another time. So question we want to answer here is, does the presence of a repellent impact the ability of ticks to detect attractant? Think about it when you go for a hike. You're spraying your repellent product, and then you're passing by on the trail, and then you have a tick on you. The tick will be exposed to the repellent that you have. What happened? How the tick respond? Is it going to be repelled right away and drop off? Is it going to stay on you? What will be the exposure over time? It will impact the tick. The tick will be used to the odors. So the second question is, has the repellent exposure a long-lasting inhibitory effect on the ability of tick to detect the attractant and the potential host? And does the pathogen impact tick ability to respond to attractant and repellents? So to answer the first question, we perform some long-term effect on attractant detection after repellent exposure. So we, first of all, we record through electrophysiological study the response of ticks to control. It was just hexane, DEET, lemongrass, and butyric acid. Butyric acid was our attractant because ticks like very stinky things. Butyric acid smells like cheese, like very strongly cheese, and they really like it. Lemongrass is a non-repellent essential oil, and of course, DEET also repel them. And as you can see, that from this recording, only butyric acid offer detectable electrophysiological response. And this is recording were done on lab-reared ticks, so infection-free. So after this, we wanted to expose the tick to the different repellent. So we use a fumigation chamber. So we did a fumigation exposure with no contact of the control, which is hexane, DEET, and lemongrass for 20 minutes. And then we did electrophysiological recording using this mounting technique. And what we found that after the exposure, only the exposure to lemongrass significantly decreased the response to butyric acid. So somehow the exposure masked the detection of butyric acid. And then after this we said, so okay, we know from the electrophysiology that there is some changing in the recordings, in the electrophysiological recording. What happened at behavioral level? So we test the response using a white bias assay. So we wanted to test basically the attraction to butyric acid before and after the fumigation. And same type of system using Ixodes scapularis females, so we work mainly with females here. And what we found is that, so the pre-exposure, so with no exposure, 73% of the ticks were attracted to butyric acid, so they were going to butyric acid. But after the exposure to hexane and DEET, they still were going to butyric acid, so no problem, they were not affected at all. But the exposure to lemongrass led the ticks to switch the arm and going completely on the opposite side. So this was quite weird because ideally, when you do Y-tube bias, you want to see the insect or the tick, in this case, to be driven towards something, towards an attractant. But in this case, the seems that after the exposure to lemongrass, ticks were preferring hexane and non-butyric acid. But then, this was right after the exposure. We asked, what happened after 24 hours of exposure? Is the effect of the exposure of the repellent last for a long time? Now, we found basically that after 24 hours of exposure, the lemongrass exposure flipped the system, so they went to butyric acid, but for hexane and DEET, they were basically going to hexane. However, these results were, if you look at the non-responding rate, it's really high. So basically, 50% of the tick tested responded, and 50% of the other tick tested did not respond. So these results are a big question mark that need to be reanalyzed and considered further. So here, just to show you with a two plots graph, so you can see just to visualize what's going on pre-exposure, post-exposure, and 24 hours exposure. And here you can see the non-responding. So basically, after 24 hours, we got 50%, 65%, and 44% non-responding. So there is for sure something going on, but it's quite interesting the trend here, where the lemongrass exposure create something going on that will lead the tick do not go towards the attractant. And then we wanted to see basically what's the exposure due to the tick. Is the fumigation of this repellent would impact the morphology, would somehow damage the sensilla located in the Aller's organs. So here we have some SEM pictures of the Aller's organ post-exposure of ticks. And we really didn't see any striking differences. So for sure there is nothing that would damage or change the morphology of the sensilla that is detectable, at least from a morphological level. So after this, then we wanted to actually test and work on ticks that are collected in the wild. So here we look like from a crime scene on a CSI mission, but yeah, we went to a research center located in the south shore of Nova Scotia, and we collected, in one day we collected 500 ticks. It was very dense, so you can just walk on this grass and then ticks just crawling on you. So very interesting. So we collected Ixodes scapularis and also Dermacentor variatis, which is the American dog tick. And we are going to use all these ticks for repeating the fumigation exposure and see basically how the bacterial infection impacts in response to repellent. And so we were going to use the ticks and store them for further analysis. So we got Ixodes scapularis, just to show you the picture. So this is the nymph, females and males. And we have performed electrophysiological response. So these are, let's say, preliminary data, because this, my research assistant is actually, as we speak, recording all the measurement. And you can see that we are seeing a very similar trend. So this is the pre-exposure, and actually this should be post-exposure. After the hexane and lemongrass, you can see that there is a decrease in the response to uric acid. And we're also doing the same type of biopsy using the American dog tick. And we are also see here a very similar trend. So here we basically, you can see that from the pre-exposure, we wanted to measure the tick response to, we have hexane and lemongrass. Here we have the tick response to butyric acid, and you can see here that there is no response to lemongrass during the exposure, the post-exposure experiment. And also we wanted to test the male, because males are kind of overlooked since the main transmission of the pathogen is associated to the female bite. So we are also interested to see how the male may respond to this biopsy. So in summary, we found that ticks are able, so the ability of the ticks to detect attractant delivered together with repellent is significantly reduced. We don't know, we are looking into the mode of action, if there is a masking effect. We know that the exposure to repellents inhibit the capacity of ticks to detect the attractant, and the exposure to the treatment reduces the activity of ticks after 24 hours. And while ticks seems to respond similarly to lab rear tick, but we need to, of course, look at the bacterial infection, because we want to correlate this with the spirochete count in the ticks. So another project that I'm involved with, also tick related, is the Canadian Lyme Disease Research Network, where different universities and Acadia and me with other people are basically sampling different area of the province, collecting ticks and sending for testing, to mapping the spread of ticks that are infected with Lyme disease, to see, the distribution of the population. And also Acadia, with my other colleagues, and other universities in Canada developed this app called eTech. It's a really cool app that you can download on the Apple Store or Google Play, which is a public platform where if you find a tick on you, you can take a picture, upload the tick, and you can get identified in less than 24 hours. But the cool part is that you can get maps where you can see in real time all the upload, and you can have a clear visualization of the density of the tick all around Canada. So it's an up-to-date information of what tick species are currently being found and all the geographical distribution. And so far, these are all the numbers as today. So you can see almost 20,000 ticks were found on humans. Okay, so thank you very much, and I take any questions.
Thank you so much. That was a brilliant presentation, and I'm sure we're going to have many questions, starting with Marcelo.
Thank you, Nicoletta. About the toxicity of lemongrass, have you checked whether this impacted survival after experiments? Because
Yes.
it might be an effect that impairs.
So we did a caricidal test using lemongrass, using the adult immersion assay. They don't die if they are immersed into it. So and we tested also the main component of lemongrass, which is citral and geraniol. They don't die. They have a very, very low mortality. So, and I was thinking, like, it's a fumigant that kills them when it's released through fumigation, because they really look affected, like impacted. They are not the mobility, the responsiveness, and so that's why it's something that after 24 hours, it's yeah, we need to look into that more closely.
Any more questions?
What about the killing behavior? I mean, some people say that they kill themselves and so on, but I have no idea what the pigs can do this.
I didn't get it.
The cleaning.
The cleaning.
Yeah, kind of.
They don't clean.
They don't clean.
They don't groom.
Yeah.
No, they don't. So what they do, there are ticks that they have aggressive behavior, and the ticks they have a passive behavior. Ixodescapularis is passive. They tend to climb up on grass when humidity and temperature are optimal, and they quest, meaning that they raise their foretarsi and they wave and try to catch volatiles. And when someone passes by, they don't jump, but they just go by. Other ticks, like the Lone Star tick, I don't know if you heard about it, the one that gives you the meat allergy, actively goes after you, as well as the American dog tick. They're very fast, so but they don't play.
I was thinking that after exposure of these repellents, they don't groom themselves, they don't clean themselves.
No, so when I do repellent assay, I usually use an arena where I put the tick inside, and then they are exposed to a repellent. They are exposed, right? What I noticed that they approach the area and then they lifted up the foretarsi. But if the concentration is too high, they kind of get overwhelmed and kind of try to run away. But they don't tend to kind of remove what they have on them. Yes.
Do you think the sustained impact of the repellent exposure on subsequent attraction to butyric acid, do you think that's likely to be central or peripheral?
You mean related to the different chemiosensory system impacted?
Yeah, I'm just wondering that when you're switching the preference, right? Switching it from going to butyric acid to going toward the hexane. Right?
Yes.
Do you think that that's likely to be peripheral or central?
Good question. I don't know. The fact that they are going to hexane and not butyric acid, it makes me thinking that they are kind of attracted to butyric acid. So I wonder if the exposure to particularly lemongrass, which is a blend, we need to remember that there are many compounds in there, it's not one like it, might create a situation where the tick is confused and is not able to process, so might be also central.
Did you do the electrophysiological recordings after the exposure?
Yes.
I just don't recall the result.
Yes, so the, I'll show this one, that it's more, so here.
Okay, so that's.
So there is a decrease in the response. There is a decrease in the response.
Got it, so there is some decrease.
Yeah.
We have time for one more question, Dan?
I wonder, is it possible there's some effect on maximum balance? Because the new measurements there, I guess it's possible to do some exposure.
Yes. So so we are currently doing electrophysiology by also chopping off the tarsi. So we did work where we chopped the tarsi, we chopped the palps, we chopped everything, and we tested on repellent. These are behavioral assay, and in this work we actually found that ticks were able to detect volatiles when they have the palps, but when they don't have the tarsi. So when you chop the tarsi, they do not detect the DEET anymore. They just go through it. But for some essential oils, and this is interesting to look also at the chemistry, particularly they were repelled by phenyl alcohol, which is a benzene ring, and it might be that this is the way how it impacted the sensilla, but they didn't get any repellents in other essential oils. So it might be that it's detected. So ideally it would be cool to do electrophysiology on the house.
Well, thank you again.
You're welcome.
Right, so we're gonna have David Carrasco. I don't think we need any introduction, we all know the bit, so.
Thank you. I would like to thank Marcelo for inviting us all, also for inviting me 10 days before the conference, especially after one speaker dropped out. It was very nice. Thank you, Marcelo. So I tried to put some slides together, so I hope they make sense. This is a project that we started about 1 year ago, and well, it is in progress. So I'm going to talk about responses to pregnancy, okay, in anopheles mosquitoes. So mosquitoes are very lovely animals as long as they don't bite you. If they bite you, they become annoying, but because of their ability to transmit parasites, they become a horrible problem. We all know that. That is a public health problem, where half of the world's population is raised from the WHO. They transmit several parasites, inducing different sickness like malaria, dengue, but you all know that. Okay. So, to avoid this, we have been using some vector control strategies that they're relying on chemicals, on, like for instance, insecticides and repellents. And what they do both is they explode the mosquito chemosensory system. Okay, repellents is here because we see it. Insecticides, well, we still don't know whether it happens or not. But at least I like to say that we explode the mosquito system because mosquitoes will go where we are, and there is where we put the insecticides, where we are. So we are exploiting their chemosensory system to control them. Okay, so this has been functioning very, very well. From 2000 to 2015, this review from Pat and colleagues, they showed that chemicals like insecticides and repellents, they have diminished, well, malaria, I'm talking about malaria, for instance, the presence of malaria in a population over the world has been reducing, and this version, 80% has been attributed to insecticides. Okay, but since 2015, now, this decline has stalled. So we are like reaching a plateau, and in some countries has increased. So there is something going on. And what we think is that what Pansacovel presented yesterday is the evolution of resistance. They are getting through these insecticides. We explained very well what were the different physiological resistance that they are very well characterized, the morphological as well, this increasing proteins or the insecticides cannot penetrate very well. But nowadays we know that there are more and more examples of behavioral resistance. We wrote a paper with some colleagues that are present here as well, saying, well, from an evolutionary point of view, what kind of adaptations could we find in the presence of insecticide control that could promote this resistance? And given this situation, imagine that you have a population of mosquitoes with a determined genetic variability. And when we put insecticides there, we will create a selection pressure. So this implementation of vector control tools will create selective pressures where some mosquitoes that are sensitive will die. Remove them from the population. That's clear. The resistant ones, they will go through, they will bite, they will spread well, I cannot touch it, maladies, sickness, or whatever, illnesses. And they will go through, no problem, because they are resistant. But then there is other mosquitoes present in the population, as Carlo was explaining very well this morning, that they could avoid these insecticides by just not getting in contact with them. And we talk about this temporal avoidance, so meaning that they switch the biting rhythms and so on, so they never get into contact with insecticides. There can be special avoidance that they bite outdoors where insecticides are not present, or the best ones are those that they don't bite humans, or if they don't prefer humans, that will go to bite animals that they really does know. But these traits can be genetically determined. These phenotypes that we see, this temporal avoidance, special avoidance, or shifts, is what Carlo is trying to determine, can be genetically determined. Okay, but also the same phenotypes could be expected if mosquitoes can detect in advance these control tools. And as well, why not? If they can detect this and they are resistant, as many mosquitoes present in nature now, they are, perhaps they could even use this information, the presence of insecticides, to say, hey, if they are insecticides, they are usually humans. And I don't care, I can go back and so on, getting a reward. So there can be a learning process. Okay. So this is what is my project, or our project. And we are studying essentially detection and whether these can behaviorally, we can find behavioral resistance caused by therapeutic plasticity, just by essentially detecting insecticides. So the most common insecticides that we have been using for several reasons are pyrethroids. And they are biosourced chemicals coming from the transcrystallinic acid, also pyrethroids. And we put synthetically, we put other ends to make them more stable. These are not very stable, they degrade very fast, they don't last very much. So by putting these groups in the end, so we make them very stable, they last for very long. Okay, so the main ones, there are a bunch of them that are used, and the main ones that are used are permetrines, betametrine, cypermethrin, and CO3. Okay, not exclusively, but the most common. Okay, so can mosquitoes detect insecticides? What we know, like from contact, it is evident that when we see them, they react to them. Is what is called repellency. So there is a sensory mechanism that they don't like. If the insecticide is more lethal at the concentration that we are applying, they will move away. They don't like it. They just fly away. Okay, which is the mechanism? Still don't know. The receptors involved, we could guess, but they are not still known at this skin. At a distance, can be they detected at a distance? Probably. I mean, there is pioneering work by Professor Gideon that have shown that in Drosophila and in Aedes, there are olfactory receptors capable of detecting pyrethrum. Pyrethrum is a mix of molecules present naturally coming from Tanacetum, from chrysanthemum flowers, which is the kind of the precursors of all these synthetic pyrethroids. Okay, and as well, I say probably because of the distance, we are using more and more what people like to call volatile pyrethroids or volatilized pyrethroids. And the effect, but behavioral, the behavioral effect we see is that when you spray this in a room, mosquitoes don't like it and they go away. So there might be something going on at the distance. Okay, but the methods that we have been using so far in malaria, the ITNs, the insecticide treating nets, and the indoor residual spray, we don't really know whether this can happen because chemically they are not really expected to be volatile at room temperature. They are very heavy molecules. Physiologically, we don't really know the receptors. I put a question mark because we know that some receptors are detected by airflow, so they are good candidates for being also detecting these synthetic molecules, but needs to be shown. And behaviorally, the evidence of spatial repellency is debated using these ones. As I told you, these volatile methods we see, but for these ones, we could even argue. And we'll talk about this. So there was a paper done by Nicoletta Faraone and colleagues that taking some data from experimental hat trials where they were testing the different mosquito nets that anti-ants that are present in the market and so on. He found that when we put them inside houses compared to control houses, you get like a kind of like a deterrence effect. I know Dr. Spitzen is not very Dr. Spitzen, I didn't use your name because I cannot pronounce it. He's not very happy about that. But when Nicoletta saw, there was some deterrence. And what he actually saw as well is that in some cases, when you wash them, if they get dirty, people wash them and they sun dry them. And after several washes, some even they become negatively deterred, which could be an attraction. So these both made us think that, yeah, there is something going on at the distance. But still, that is really weird because of the reasons I told you before. So we went further. And we went to, well, it's actually PhD students, somebody is sitting behind them. And went to Burkina Faso and we tried to collect pyrethroids from nets and see what happened. We followed the protocol very well, very good protocol from Yoshida 2017. And we collected the others from a room where they was present for a couple of days at different distances. And we wanted to see if we were able to capture permethrin. In this case, it was a net commercialized as MAK'IT, which has 2% permethrin at PBO. Okay. So we collected these others for 24 hours, and we are 48 hours, and we analyzed them. And to our big surprise, permethrin was there, unwashed, and sun-dried as well. Okay. This is still going on, the analysis, because we were very focused. We were looking at the chromatogram to look for the mass spectrum of permethrin, whether it was present or not. I was surprised when we found it. So we can capture permethrin is present in the room air. Okay, now we are trying to identify the other compounds because there are plenty of integration compounds. Some of them have been shown to be repellent in mosquitoes, and we want to quantify this, how much it is the benefit for being behaviorally relevant for mosquitoes. And also we are performing this with different commercial ITMs with different insecticides. If you are very sharp, you will say like, hey, here the permethrin goes like, well, it goes out at 20 something, and here it goes at 16. This is no problem because we change this method. We're actually trying to get optimize the methodology, and as I told you, this is very preliminary. But what we were clear and happy is to find the perfect. Okay, so it is present in air, in room air. Later, we wanted to ask the mosquitoes in a system that could mimic a little bit the conditions that we find in the field. So for doing that, we put a mosquito inside a tube and at the distance of about 1 centimeter, not very long distances, but we wanted to know whether this could work or not. So it was really a proof of concept, seeing like something is happening. And we use a stimulus controller to throw a puff of air during 30 seconds. And this air was getting filtered by charcoal, and then going down in a bain-marie with, we could regulate the temperature of this molecule. Why do we want to check the temperature of this molecule? Because usually we do the experiments at room temperature, and in field conditions we don't find these temperatures. You can have 25 degrees in some places, and in some others, 25 degrees you find it perhaps, I don't know, like in winter if there is any winter. So we use 2 temperatures here. 25 degrees would be room temperature, but also 35 degrees that could happen, and we can see, we can experiment these temperatures, for instance, in Burkina Faso or in Western Africa or the southern side. Okay, so we took into account these physical-chemical properties of compounds, that usually they are, not really good. So what we saw is that using Kisumu is a strain, laboratory strain, which are sensitive to insecticides, that we could see a different, it's not really concentrations, it's masses that we put. So that we see that there is, I didn't tell you what we were measuring. Actually, when we were puffing, the mosquito was just quiet, standing on the net, just very happily standing there. So we approach the tube and then we puff. And what we measured is whether he liked it or not, he escaped or not. And it was a very clear response that we don't like it, or I like it, or I don't care, actually. Okay, so what we see here is like at a very high concentration, this concentration, perhaps this high concentration we used is the equivalent of what is in on 1 square meters of these mosquito nets. Take into account as well that not all the permethrin that we find in mosquito nets is bioavailable. Usually they are embedded in these tissues, and with pass of time, the permethrin is migrating to the surface is what the mosquito takes into, gets into contact. But in any case, we were completely blind, so we started with this quantity, and we saw that they really didn't like it, regardless of the temperature. And then we did this, we scaled down to see what was the limit. So we saw that at 0.4 there were still differences, at 0.04 there were still differences, and then we reached a plateau that we didn't find any differences. Okay, yes. So I must say as well that these figures, the error bars are 95% confidence intervals in all my figures. So they are very easy to interpret. So if they don't overlap, there is no difference. Now, if they overlap, there is no difference. If they don't overlap, there is a significant difference. Okay. So we see that there is a little effect of temperature, but not as much. If you have seen some piece poster on D, which is pretty much the same, there we have a very clear effect of temperature. Not in experiment. There is for sure. But, okay, so we also tested Kisumu, the same strain, but this one has a physiological resistance, which is the knockdown resistance. Okay, so we did the same, exactly the same. They didn't like it, but if we compare it, even though we can barely compare, if I go back and forth, you will see that the sensitive, the real, we could say like escaping behavior, we find it at this concentration or mass of product. And this here, in the resistance, it seems that has shifted a little bit, so they are less sensitive to this. But this has to be taken, because we cannot really compare. Okay, so after that, we wanted to ask, like, okay, this is Kisumu strain, has been treated in the lab for many, many years. So we went to the field and checked. We collected larvae. It was actually somebody that did that. we collected larvae, we did experiments, and then PCR them to see the species. So, and in this year, he did the same experiment. So what we see that all of them, in these conditions, like highest concentration at 35 degrees, all of them escape. So there is something going on as well. Okay, then we started saying, okay, we see a behavioral response. We want to know which organ is involved, which sensory organ is involved in this detection. So we started chopping the different organs. In this case, we cut the antennas, and what we see is like antennas don't play any role on this behavior. So we're like, okay, we were surprised. We were thinking that perhaps antennas would be the major contributors to this behavior, but not at all. Okay, so we had to go through all the organs. We went to the palps, it was exactly the same, it went down, not different. So we went to the proboscis, perhaps the proboscis has some receptors there that can determine nothing until we went to the legs. The legs, we recovered them in dressing because if we cut the legs, it's very difficult to record the behavior, especially the scaling. So we put some socks on these mosquitoes, and we see that the kind of like this behavioral response towards permethrin was completely abolished. So we thought like, okay, perhaps it's the socks that really go back this. So we checked another positive control, which would be xenominol, which we know that it acts it is detected not only through antennas but also antennas, and there was absolutely no problem for them to scale. So socks didn't beat them to scale, it's just they didn't detect this permanently. Okay, we were not expecting this. I mean, we didn't think that molecules that are kind of like volatilized in some sense can be detected through the Darcy. We always thought that Darcy go, when they approach, they just land on a place, they start tasting, but not from distance. Okay, so, and when we saw this result, immediately we thought like, okay, there's this paper from Dennis and colleagues from Leslie Warsaw's laboratory saying like, Aedes aegypti use the legs on contact. Here, there is no contact with the source. It's basically all the volatiles that are right there. So we started doing this on physiology. Okay, so to do this, we went through all the organs as well, but the proboscis. And what we found is the antennas respond to this, respond to permanently. Very volatile. And we have a system, we created a system that we can do electrophysiology and stimulate or send the others at different temperatures. We can regulate the temperatures of our cartridges when we are loading with the chemicals. So we tested both temperatures as we did with the behavior, and we see that the antennas are responsible of detecting the permitting, but the behavioral response is not associated through the antennas. We went to the palps and we did electropalpograms, and we see that there is absolutely nothing. I didn't explain. So basically, these are still confidence intervals. If they touch 0, they are not significant. As long as they are up 0, they become significant. All right. So here, yes, I didn't say that. Here we use a positive control acetophenone, which is known to be detected by the antennas, and we use it before and after to check, because as insecticides can be neurotoxic, to check that the antenna was still okay in the end of the experiment. So the palps, you see, there is absolutely no response. The positive controls, in this case, was 1 ampentriol. They were both responding, and we went to the tarsi. Here we use ammonia as a response. We see that at certain concentrations and at certain temperature, it's a matter of volatilizing more or less the molecules, we have responses. We check as well that the scale are not the same. And this one, the response to ammonia was huge. It was huge, and these responses were very, very, very clear. Okay, so we have antenna and tarsi that can detect these compounds from a distance. So to summarize the results, from chemistry point of view, permethrin from methane can be found in air. That is clear, we find it. What is good to know now is the degradation compounds. We haven't checked them, and we believe they play a major role in this, what we all see, well, what we see in the behavior and what we see as well in the electrophysiology. We need to identify these degradation compounds. We have a guess what they are, but we need to find them and identify them. And what about the other pairings? Can we collect also other pairings? From the behavioral point of view, escape responses are only elicited by the information coming from the past. Okay. And yes, I forgot to say that degradation compounds usually pyrethroids degrade either through UV. When mixed with water, you speed up the process. And after Magali's talk, I thought like, hey, it'd be interesting to check also because of the double bonds they can break and ozone is present everywhere, especially when it's hot. Okay, so finally, the physiological. There are two sensory mechanisms of detection so far. One is the TARC, which is clear. The antenna, which is redundant. It doesn't seem to elicit anything, but they can do it. Next, which receptors are these? Olfactory receptors, gustatory receptors, ionotropic, otrix, or other, any other receptor. So now it's the coming work is going to be focused on finding this. I don't even expect that it's only one. I think there are several of them that are involved. And I'm very interested in finding this, and this. So to come in next, okay, we found things in the lab with different concentrations, very high sometimes. We force it to volatilize. We need to quantify how much is emitted by the tenants and so on. So we need to know if everything works in the field. Because if it doesn't work in the field, we don't see any response in the field, behavioral resistance doesn't exist. Yes, it's a good test, it's a good proof. It works this way, the mechanism. It can be unfit, but perhaps they don't do anything about that. So we'll go to the field and try to check whether sensory detection of insecticides can elicit or can evolve this temporal avoidance, spatial avoidance of host shifts. But also, we're gonna go as well to check the fun part, whether the mosquitoes can learn about this. They have receptors in the antenna that detect, but they don't do anything, but they detect. So meaning that if they detect an odor and they get into contact with the insecticide and they have this excitor repellency effect, they will burn themselves, they can perhaps learn and say like, okay, I smell the permethrin and then I got a shock. I won't touch it anymore. So they will prevent this avoidance will be very clear after some learning process. Or the other way around. It could be as well that the positiveness, the stimuli that they get, the reward is not a reward, but it's not a punishment, but it's a reward because they don't care, they are resistant, they can go even they sense the permethrin, they manage to feed. Or perhaps they can use permethrin to say, hey, where there is insecticides, I have more probability to find humans. I know that it's, but it's fun to test. And also, what about volatile pollutants? Actually, very interesting, because perhaps using the items, well, the insect treatments on the indoor residual spray, perhaps we don't find anything like this. And now, these volatile pollutants like their own. Spatial repellency, we put them in a place, mosquitoes go away, they even die, they're very effective. But if they detect this, perhaps we will favor much more on the apparition of this behavioral resistance. I would really like to thank all the people in our group. Anna, Sheila, Cedric Penetier, which helped very much in this project. Also our collaborators in Burkina Faso from the RSS, Sergio Manga, again, our collaborators from Sweden that we work with the receptors, olfactory receptors, Ricard, Inel, and also Aman Bonaventure, because most of the, I can tell you, Sweden on Earth, but they have found NOR that might respond to permethrin. It needs to be confirmed. And it was thanks to the work of Aman. And especially, I really want to thank these two persons here, our PhD student, Francisca Wu and Ivan, that they did most of this work very, very, very well. I announced as well that we were recruiting a person for next year, so if anyone wants to join please contact me. It's gonna be on the fun part of the experiment, also very risky, you know. Need to put it on the balance. And I would also thank the funding agencies, the key initiative, risk vectors that settle and this encourage this to happen. And then this project was funded by the Agence Nationale de la Recherche and also the IRD for funding the PhD thesis of this. And I thank you all. Thank you very much.
Thank you so much, David. We have, I can see someone wants to ask something, yes?
Yeah, but maybe we should talk tomorrow, but there's so much to say and show you. This is really.
You know more than me.
Sorry?
You know more than me. That is good.
Well, I want to save you a lot of money, then you don't go to the field because we did it.
Okay.
No, but it's the same year as the 2017 the other paper came out. Anyway, there's a lot to do in the field, so you should go and more to learn so we can learn from this. So what we did, we had the different nets inside our house. The parameter net and the winter net, but I think the main point I want to make to the group is that the combination of human odor inside the house is crucial here.
Yeah.
And there was an earlier paper by Omondi et al. In 2012 that shows us for the first time that mosquitoes can overcome the repellent effect when there is a realistic odor behind it. And so that makes sense. So I think what we did, we filmed the entrance at the deep to see whether mosquitoes would still go in when there is a net there, and there was no difference between the entry net and the treatment box. Yeah. There were no, I have to.
Oh, how long did you have the nets there?
Just overnight.
Overnight.
Yeah, and then we switched the nights between the experimental setting in Kenya.
Yeah.
So we switched every night.
We should do this, leaving the net for 1 month.
Yeah, but the other thing, we had a synthetic blend inside because it was standard over time. But there is comments on that, and we work with volunteers. Volunteers are also not stable over once. So that's the other. Yeah, sure. So that's complicated. It would be nice to repeat it. But I have very many doubts that it will be repellent at that. I hope it's proven wrong. Yeah.
Is it
You
interesting
can look at
discussion?
movies tomorrow.
Yeah, sure.
Yeah.
Perfect. We have time for just a couple more questions.
And so how do you explain the experimental habits?
It didn't.
Yeah, but there are so many studies.
Oh yeah,
demonstrating that there is a reduction in entry. Well, there is a reduction in the number of mosquitoes recovered in experimental habits.
Yeah. So we discussed this in the Anthropocene paper, and I think that discussion is not finished. But of course they die. Like the proportion is dying somewhere thousands. Ones are dying faster. Others are not, and for parameter not. So we looked at how many exit by observing and how many do enter. For entering, there is no difference. It's proportional. If they cannot feed because there's a bad net and a human, they go out again. So maybe that's where the difference is.
Well, but the experimental habits do control for this because they have slits that avoid the exit from the entry point. But then they have verandas where they do go out. And if you correct for the totals, we just have to control, of course. Then you demonstrate that overall you have less mosquitoes that have entered.
Yeah. So there is something going on there that would be nice to also film inside and see what's happening. And that is more difficult than what we do. So maybe that's where you can spend the money.
We have time for one quick question. Yes.
Yes, I was just you remember on your last slide. Why you address work on chemistry? Why not working with more volatile products, especially the ones that are used for partial repellent control tools?
Yeah.
Methylfiltrine, phosphofiltrine, we know they work.
Yes.
We know they have their mode of action is really to prevent the mosquito coming in houses, and we have more evidence also in field that it's working well, even with randomized control trials. So well, clearer response about the 62 repellency, why not also targeting this particular?
It's planned. It's planned in the proposal, it's planned in the, well, in next years, and going to the clinical test all next. But about this special repellency using sorry, I'd like to just say there was one study saying like, I mean, this control method where you volatilize this permanently indoors, it works very well. They have shown that it works very well even in kids hospital, but they found that it deposits as well everywhere. I mean, you can find these insecticides everywhere, and that was a danger for toddlers, which are like walking on the floor and they put the hands, even though pediatrics are known to be not so very harmful. So I wonder what's going to happen for that pediatrics. Thank you.
Well, thank you so much.
On there. I just wanted to first thank the organizers, Marcelo and David, for inviting me to share our work with you. I really wanted to be there with you today. Unfortunately, my flights were canceled and I wasn't going to make it there in time. As the French say, c'est la vie. So I'm going to be talking about mosquitoes today. And in particular, my lab works on, laser pointer on, hold on a second. Let's see, laser. Okay, there you go. I'm going to be talking about Anopheles mosquitoes. That's what my lab works on. Some have called these Anopheles mosquitoes the most dangerous animals on Earth because they are the vectors for a Plasmodium parasite that gives rise to the disease malaria, which kills around 450,000 people every year. There are also a number of other pesky mosquitoes. They are also vectors for a number of other diseases. And what all these mosquitoes share, what they all have in common, oops, start my video. Hold it a second. Gotta click a button here. Hold on a second. Can't click buttons when your laser pointer is on, apparently. Start. All right, there we go. What all these mosquitoes share is that they use their sense of smell to find us. And so that is essentially what my lab is doing. We're targeting the mosquito's sense of smell to stop them from finding us and stop them from biting us. And everyone has actually done this experiment already to use the sense of smell to target a mosquito's sense of smell. And that is essentially mosquito repellents. That's what mosquito repellents are supposed to be doing, is targeting the mosquito's chemosensory systems to stop them from finding us and biting us. And when we started this work, we were pretty amazed to find out that we know very little about how these mosquito repellents work. We've been using them for many years, but we have very little understanding about what these mosquito repellents are doing. And so that's the focus of my talk today. So let me just give a quick primer on the mosquito olfactory system. So here we're looking at a side profile of an Anopheles gambiae female. And I've just pointed out the major olfactory organs of the female, the antenna, which contains most of the olfactory neurons, the maxillary pulp, and the labella. If we were to zoom in on the antenna, you'd see that it's covered in these hairs, special sensory hairs called sensilla. And these sensilla, if you do a cross-section, you'll see that those contain the olfactory neurons. So the way this works is that odorants in the air will pass through the pores that are in the sensilla, reach the odorant receptors that are on the dendrites, and activate these olfactory neurons. So our goal is to essentially genetically target this mosquito's olfactory system. So how do we do that? So what we've done is we've developed new genetic tools for the mosquito. So we've developed these tools, these genetic tools called the Q binary expression system. So we originally developed these in Drosophila and we brought them over into the Anopheles system. So I just wanted to do a quick explanation of how this system works. So this is a two-component system. So the first component is a transcription factor called QF2. We originally got this for Neurospora. So it's an exogenous transcription factor that doesn't normally exist in mosquitoes. And in our case, we're expressing it off this Orco enhancer promoter. So Orco is a gene that's expressed in most olfactory neurons. And so in this case, QF2, this transcription factor, will be expressed in most olfactory neurons. So QF2 will bind its enhancer called QAS and turn on whatever you want. And so in this case, we're turning on GFP. So the way this works, let me just turn off, sorry. I got this chat window here that is in the way. Hold on one second. Just close that. Here we go. Laser point back on. So the way this works is that we have one transgenic mosquito that contains QF2 driven by the orco promoter, and we have another transgenic mosquito that is driven by the QAS GFP. So this reporter by itself doesn't have any expression. So you cross these together in the progeny, you'll now get GFP expression wherever orco is expressed. And one of the advantages of using a binary expression system like that is it can also amplify the signal, so the GFP signal will be very bright. And also it's very versatile, as we'll talk about a little bit later. So this is what it looks like. So here we are looking at a light sheet microscope view of the Anopheles gambiae mosquito with this combination of genetics. And so you can see that the antennae are beautifully labeled. So you can see the olfactory neurons in the antenna, the olfactory neurons in the palp, and then the olfactory neurons in the labella. So what's the most important part of this, though, is that this indicates that we now have genetic access to these olfactory neurons. And so we can actually then start asking the question, how are insect repellents affecting a mosquito's sense of smell? So I wanted to mention that this work that I'm going to talk about was mainly done by a very talented postdoc in the lab, Ali Afifi, who is going to be starting his own lab in September. So there are two broad categories of insect repellents. The first one are the man-made repellents. The gold standard by far is DEET. So DEET is an effective repellent. There are some downsides of DEET. One, you have to use it at a relatively high concentration, usually 30% or higher for it to be effective. It is also a plasticizer and a rubbercizer, so it can dissolve plastics and rubbers. It is slightly neurotoxic, so you don't want to get it on your lips or in your eyes. So there are some other synthetic man-made alternatives. So there is IR3535, which was developed by Merck, and Picardin, which was developed by Bayer. If you want not a synthetic, there are a number of natural products. So natural means they're usually derived from a plant or some kind of natural product. And so the ones that are most commonly used, which we heard a little bit about today, is oil of lemon eucalyptus, and the active ingredient is called PMD. And there are a number of other natural products that are on the market. And so, for example, lemongrass oil. So what I'm going to be talking about are these particular repellents. And so again, what these are doing, how these are affecting the mosquito's olfactory system is not well understood. So that's what I'm going to be talking about today. So let's first, maybe take a step back and figure out where do these things come from in the first place. And so let's just talk about the story of DEET. So DEET was identified in a screen that was really as a result for World War II. A lot of American soldiers were going over to Europe and Africa and essentially getting ravaged by malaria. So the United States Army teamed up with the USDA and realized they needed some way to protect soldiers from mosquito bites. And so, the original intent was to identify something that could protect soldiers from Anopheles mosquitoes, but raising Anopheles in the United States is more challenging. So what they did is they did a screen with Aedes aegypti mosquitoes. And so what they did was they essentially had a box that you see here. They had 2,000 to 4,000 mosquitoes, female mosquitoes, in this box. And then they would have volunteers put a chemical on their arm, wait a certain amount of time, usually like half an hour, 3 hours, and then put their arm in this box with all these hungry mosquitoes, and then see how many bites did that person get. And so the final output was, did you get bitten or not if this chemical was on your skin? And so DEET was essentially discovered from this type of screen. So we can see here that, DEET was discovered because it has this activity. It prevents mosquito bites, and not because we had any idea of how it was working. And so DEET for the general public was available in 1957, and after about 60 years, the function of DEET was still not clear. So why is that? Why, after all this time, we didn't have a good idea what DEET was doing? And the main reason is that it's really hard to visualize the odor responses in mosquito olfactory neurons. And so, for example, just on the antenna alone, there are 600 scintilla, 600 of sensory hairs, about 1,200 olfactory neurons. And so in order to figure out what DEET is doing, you'd really have to figure out the activity of DEET towards these 600 different scintilla. Now, it is possible to record from scintilla, so you can stick an electrode into these hairs and then record the electrical activity of these olfactory neurons in response to odor. However, that's very challenging to do, and these hairs are fragile, so it's pretty easy to break them. So to do this with all 600 scintilla across the entire antenna is pretty much impossible. So the strategy we had was we'll introduce genetic tools into anopheles that allow us to directly visualize how these olfactory neurons are responding to odors. And so this is, again, the advantage of using that binary expression system. So before we were just using GFP. Now we can switch it out and use a different reporter. In this case, a modified version of GFP called GCAMP. So GCAMP is a modified GFP that's also a calcium sensor. And so whenever there's an influx of calcium, this GFP will fluoresce brighter. And so you can be used as a proxy for neural activation. So whenever an olfactory neuron is activated, there's an influx of calcium, and this particular version of GFP will fluoresce brighter. So shown here on our right is our relatively simple prep. So here we have a female mosquito shoved into a pipette tip, and then we have her antenna pinned down on a slide. And so all of her olfactory neurons are exposed this way. So we can essentially just puff on an odor and visualize how the olfactory neurons are directly responding to those odors. And this is a live prep, so the mosquito is alive still. And so what we can do is we can actually look at those repellents. We can puff these repellents on and directly see how what these repellents are actually doing to this mosquito's olfactory neurons. So shown here is an example of puffing on lemongrass oil, a natural repellent. So hopefully the video will play. Here we go. You'll see like the puff of odor there with the little green bar will indicate that we puffed on lemongrass oil. And I hope what you can see is that there were 4 neurons that were activated. There was these 1, 2, 3, 4, and we can diagram that as a change in fluorescence to delta F. So that's those neurons that essentially fluoresce brighter, ones that were activated. And you can see that there were 4 neurons. You know, 1 neuron turns on and then turns off, and then there's 3 other neurons turn on for a little bit longer after the puff is gone. And so what this essentially indicates is that lemongrass oil, at least for this one, segment of the antenna, is activating 4 neurons. And so one or maybe a combination of all these 4 olfactory neurons are sending a signal into the mosquito brain to stay away from this particular odor. This particular odor, for whatever reason, is bad that the mosquito should stay away from. The activation of these neurons are sending that signal into the brain. And we saw that for all the natural repellents we tested 15 different natural repellents. They were all activating discrete and reproducible subpopulation of olfactory neurons on the Anopheles mosquitoes. So we think that's what these natural repellents are doing, is activating these neurons. These neurons are sending a signal into the mosquito brain to say, stay away from these odors. The real surprise, though, came when we started looking at DEET. And so I'll show what that's on this slide. So I'm going to first show a positive control. This is a response of the antenna to 2-acetylthiophene. It's an odor you might find in your skin, an attractive odor. And you can see that when you puff on this odor, that a lot of the olfactory neurons are activated. And so the surprise came when we also puffed on DEET. And so we were expecting to see a large number of olfactory neurons were activated, but instead we actually didn't see anything. There were no olfactory neurons that were activated when we puffed on DEET. We thought we were going to see like what we saw for natural repellents. There were some populations of DEET neurons that were activated. So we thought, well, the way you use DEET is you mix it on your skin. So what would happen if we mix DEET with this odor, this 2-acetylthiophene? So you can see that when we mix these two together, this mixture, there also is no response. And so what we can see then is that when DEET is mixed with this odor, this 2-acetylthiophene, which was originally very strongly activating the olfactory neurons, when DEET is mixed with that, you no longer get a response. So, from these relatively simple experiments, again, we're looking at almost all the olfactory neurons on the antenna. We can conclude that, DEET is not strongly activating these orco-positive olfactory neurons in the Anopheles mosquito, but what it is doing is preventing the ability of this odor, this 2-acetylthiophene, from activating the olfactory neurons. And so this is a process we call masking. So DEET is masking the ability for other odors to activate the olfactory system. And this is just to drive that point home. So here we're looking at the different olfactory organs, the antenna, the maxillary pulp, and then the labella. Here we have a positive control. So this is just a response of these organs to benzaldehyde. So you can see there's a number of olfactory neurons that are activated to benzaldehyde, even in the pulp and the labella. However, if we puffed on 100%, we couldn't get any more DEET. We puffed on 100% DEET and 100% IR3535. There was just no olfactory neurons that were activated in these olfactory organs. For picardin at 100%, there was slight activity to picardin in the pulp and the maxillary pulp and the antenna, but this was only at the highest concentration. So we reduced the concentration, we didn't see anything. So from these experiments, we concluded that these man-made repellents, these synthetic repellents, DEET, RA35, 35, and Picard are not strongly activating Anopheles olfactory neurons. We can also found that there is a dosage dependence masking by DEET. So what we're looking at here, so you're looking at the response of this one segment of the antenna to octanol. It's an attractive odorant you can usually find in your breath. So in this case, you can see that there were 4 olfactory neurons that were activated to octanol. And so what we're going to do is we're going to see now how is the activity of these olfactory neurons affected when this particular odor is mixed with different increasing concentrations of DEET. So what we're seeing here on the y-axis is a change in fluorescence, essentially how strongly those olfactory neurons were activated. And the different colors indicate different animals that were tested. So for example, here we have like one olfactory neuron that was at a certain activity level when activated by octanol. When that octanol was mixed with DEET, that same neuron had a slightly lower, not greatly affected response. However, if you start increasing the amount of DEET that you're mixing in, so for example, here we have, this one particular neuron responded like this to octanol. However, if the same neuron responds to octanol when it's mixed with DEET, it's essentially gone. And so we think that this, this effect of this stronger masking effect might be also one of the reasons why you have to use relatively high concentrations of DEET in order to engage this particular masking process. And so for the interest of time, I'm not going to go into all the experiments we did to figure out how this masking was working. So I'll just bullet line, give the bottom line. So the masking is working, in this case for Anopheles, by reducing the volatility of other odorants. And we weren't the first ones to identify this for DEET. This was originally identified by Walter Leal in 2008. So our model then for Anopheles mosquitoes for DEET is that, usually the mosquito is attracted to a number of odorants that are on your skin. So mosquito is attracted to that. When you place DEET on your skin, you're kind of masking those odorants or keeping them trapped onto your skin. So that mosquito will no longer really be able to find you. You really don't smell human anymore. And so it'll go find somebody else who perhaps isn't wearing DEET. So I think the bottom line is always to bring a friend with you that is not wearing an insect repellent. So we kind of think of this as like, the invisibility cloak, that when you put DEET on your skin, you're kind of hiding yourself from the mosquito. And so what's happening here is really a loss of attraction due to decreasing the volatility of attractive odorants. So at this point, you're probably thinking, wait a minute, I thought DEET was a repellent. It even says so right there on the can. It says insect repellent. So maybe, it's still a repellent. Maybe it's just that we're just not seeing the olfactory neurons that are activated by DEET. Maybe it's able to act as a spatial repellent, odor repellent in some other way. So we decided to test that directly. And I should mention that a lot of the assays that are currently used are not directly testing the response of that odorant as a repellent odor alone. So for example, the assay we talked about that was used for discovering DEET, this Arman cage assay, that is a huge amount of mosquitoes in a very small space, 2,000 to 4,000 mosquitoes in a very small space. And so, and the final outcome, what they're actually measuring is how many bites did that person get. So we think that this type of assay is really biasing towards contact repellency. You're looking for chemicals that mosquito touches it, they're not going to like, and they're not going to stay around to bite, and perhaps to odor repulsion to some degree. And the other type of assays, for example, is something like this, a 2-choice olfactometer, where there's 2 different choices, 2 different odor sources that the mosquito can choose from. And so what's usually done, you have a person put their hand in one choice, and then they put their hand in the other one with DEET on top of it. And so this is not, this is not testing DEET alone. What this is really testing is how the attraction to a human odors are affected by the repellent. And so we decided that we needed some way to test this directly. So we came up with a very simple assay we just called close proximity response assay. And so basically the way this works, you have a single female mosquito, and you put her in a cage, and this is netted cage, and you just allow her to come to rest. So after about 5 minutes of just resting, you then bring the odor to wherever she's resting. So bring the odor within 0.5 centimeters, and you just watch what she does over the course of 30 seconds. So, if she's repelled by this odor, she'll fly away. If she's not repelled by the odor, she'll just hang out where she is. So this is what it looks like for DEET directly. I should mention that what we're looking at is individual animal assays. So we did this 30 times, so 30 individual experiments. And what we're graphing here is the proportion remaining over the course of 30 seconds. So for example, here for paraffin-oil or negative control, you can see that of the 30 mosquitoes, it looks like one flew away around the 29th second. This is what it looks like for lemongrass oil. So lemongrass oil is very effective odor repellent. You can see that most of the mosquitoes have flown away within 2 to 3 seconds. And the same for PMD. It's a very effective Anopheles mosquito repellent. Most have flown away within 5 to 10 seconds. So what about DEET? So this is the response to the odor of DEET. So this is 100% DEET. They essentially ignore it, and it's not statistically different from paraffin oil. So this would indicate that the odor of DEET alone is not repellent to Anopheles mosquitoes. And that was the same for picaridin and the same for IR3535. So, this indicates that, the odors of these synthetic repellents, DEET, IR3535, and picaridin, are just not repellent to Anopheles mosquitoes. But what about other species of mosquitoes? So I just wanted to point out that in the last common ancestor between the Colchicine mosquitoes, the Aedes aegypti mosquito, and Anopheles mosquito was about 150 million years ago. Just to put that number in context, the last common ancestor between humans and mice was around 80 million years ago. So 150 million years ago is a long time ago. And so there has been plenty of time for the olfactory systems to diverge between these two different mosquitoes. So that just means, whatever we find in Anopheles mosquitoes doesn't necessarily mean it'll work the same way in Aedes, and vice versa. All right, so let's look at Aedes. So the same assay. So here, paraffin oil, the negative control. Lemongrass oil is also an effective spatial repellent against Aedes aegypti mosquitoes, as is PMD. It's not as strong, not as effective as it was against Anopheles mosquitoes, but the odor still is repellent. So what about DEET? So this is the response to DEET. So DEET towards Aedes aegypti mosquitoes is actually repellent, is a spatial repellent, is an odor repellent. So this would indicate that DEET, that Aedes aegypti mosquitoes very likely do contain odor receptors that can detect DEET, and this is sending a signal to the brain of the Aedes aegypti mosquito to stay away from this particular odor. Picaridin, same as for Anopheles, is not a spatial repellent, and neither is IR3535. So this is our model for olfactory models for DEET. So Anopheles, really it's just masking. That's the main thing, that odor, what it's doing to the odors of DEET. And for Aedes aegypti, it's the repellent odor, so it can repel them that way as well as masking. And I should also point out that DEET can also act as a contact repellent. This was work that is found in Aedes aegypti and Anopheles gambiae. And this kind of makes sense because the way DEET was identified in the first place is in this Armand cage assay, which we think really does bias more towards these contact repellents versus spatial repellents. And so, as I mentioned, these man-made repellents, they engage masking, and the natural repellents are engaging activation. So what would happen if you mixed the two together? Would the masking ability of these synthetic repellents essentially cancel out the ability of the natural repellents to activate and repel mosquitoes? So we decided to test that. So here we're just looking at the neuronal activity towards these two natural repellents. So here's the response to PMD. You can see there's a number of olfactory neurons that are activated. And here's the neuronal response to lemongrass oil. There's also a number of olfactory neurons that are activated. So when we mix PMD with DEET, so at 30%, you can see that the neuronal activity goes down. So this would suggest this particular mixture of PMD and DEET will be maybe less effective repelling mosquitoes as the PMD by itself. However, what's really interesting is that if we did the same thing with lemongrass oil, that the neuronal activity was really not that changed very much. So it looks pretty much the same. So DEET is not really able to mask as strongly lemongrass oil. So this, would suggest that lemongrass oil, the components in lemongrass oil that are activating these olfactory neurons, are very efficient at doing it. Perhaps you need a very little amount, very low concentrations of these odorants to activate these olfactory neurons. And this would suggest then that this combination might be a much better insect repellent against Anopheles mosquitoes. One, it can act as a spatial repellent, and it still has that masking effect. So we decided to test that. So here we're looking at the behavior. So we have paraffin oil as our control, and here is the response to lemongrass oil and to PMD. When you mix PMD with DEET, you get decreased response. So that is what the neural activity suggested, that this wouldn't be as effective as a spatial repellent. What about if we mixed lemongrass oil with DEET? The neuronal activity suggested that it would still work very well as a spatial repellent. And sure enough, that is what we see. So you can see that it looks pretty much unchanged. So, this would suggest that this combination of DEET and lemongrass oil could be a very effective combination for using against Anopheles mosquito. And what's, this is interesting is that we can essentially use this strategy that we can look at the neuronal activity of the mosquito itself to predict and identify effective repellents or mixtures. I'll stop there. So I just wanted to thank my amazing lab, in particular Ali Afifi who conducted most of this work, and my funding sources. And I'd be happy to take any questions.
We have time for a couple of very quick questions. Do we have any from? Yes, so we have one from Carlo.
Yes, first of all, thank you. That was an amazing presentation. Very, very clear, very fascinating results. I loved it. Just one word of caution about this use of natural repellents combined with other kinds of repellents. In a practical way, when we extrapolate results from the lab to the field, what matters at least as much, if not more, is how long the repellent is going to be efficient. And that's one of the main limitations for natural products like lemongrass oil. So just to say that perhaps it should be interesting to see what's the range of effectiveness through time.
Yeah, I think that's a great point. So we think one of the reasons why DEET is slightly longer lasting is because it's very low volatile, and that's why it can last on your skin. And one of the reasons why natural repellents don't last as long, you need to reapply, is because they're more volatile, so they're not going to last on your skin. So, the interesting aspect is that if, we take a natural repellent and mix it in with a low volatile odor like DEET, it might have the effect that it reduces the volatility of the natural repellent. So you might be able to actually have that natural repellent last a little bit longer because it's not going to be as volatile. And it seems that, lemongrass oil, whatever the components are in there that are effective, don't really need to be used at such a high concentration. So we might be able to get away with even having a lower volatile version of lemongrass oil. So it might actually last longer. But you're right, this has to get tested in the real world.
Perfect. We have just time for one more question, but I will invite everyone else that has a question to just put it in the chat. I'm sure that Chris will be happy to answer it. But yes, go ahead.
Thank you for the presentation. I just want to comment about the concentration of lemongrass. So it clearly works when you use a 30%, but I think we need to also consider the application on their skin. And I know that lemongrass oil is a sensitizer, and you cannot apply more than 4% because it's going to induce, rash and sensitization. Now, I'm familiar with the Health Canada regulation because I'm in the process of registering a product. So I know that there is more restriction. I don't know the EPA, but the other comments were about in the field when we combine and we use natural product. Unfortunately, we need to also look into the potential effect that we have when we apply on the skin. But this is amazing work. Thank you for sharing with us.
Yeah, we didn't test lower concentrations. The reason why we tested 30% was because we were trying to see how much of the masking was going to happen with DEET. So we'll try lower concentrations to see what, kind of titrate it basically to see what was the lowest concentration we can actually use to see neuronal activity of those neurons. And I think what we'll find is that it'll probably be in that range of 1 to 2% of these odors, these chemicals, in order to actually have an effective repellent. which is probably why, we use it 30% is too high, absolutely. And so I think that is probably one of the reasons why it can still be effective even when mixed with 30% DEET.
And you can always encapsulate the oil to protect them and then to reduce any thermal interaction.
Right.
And keep it like a higher concentration.
First, I would like to thank you once again. We have a few more questions, so please be on the lookout in the chat and your emails. I'm sure people will contact you. And please join me to thank him once again.
If you want to avoid mosquitoes, you have two main common ways. The first one is to be around someone who is more attracted than you to the mosquitoes. But if you don't have this guy, so you need to use DEET, which is the most common insect repellent. And over here you can see DEET inside Israeli product. It says enough jungle. So DEET target gustatory and olfaction receptors. And while trying to understand the mechanism behind the olfaction repellency of DEET, Hoofput and Dickens used TEBC, which is two electrode voltage clamp system. So what they did, in this system, you basically take mRNA, for example, OR8, and inject it inside a Xenophus frog oocyte, and then the egg expresses the odorant receptor. You can measure the electrical current like in this trace. So what they did, they exposed OR8, which was expressed on the oocyte. So they delivered octanol on OR8 and they saw an activation. But when they mixed it with octanol, they saw an inhibition. And they thought that it might be due to non-target sites, which the DEET bond with. And it's actually a nice correlation between the talk I give now and the talk that Chris Potter gave. So it might be the reason that OR8 is not activated, but maybe there's another reason. And surprisingly, they also found that DEET can activate different ORs. For example, they show that DEET can activate OR2, which is responsible for the oviposition cue indole. So, based on these results, Esther Yakir, our lab manager, hypothesized that the chemical similarity between indole and DEET might create also inhibition in OR8 and might also inhibit human host traction behavior. So, Amir Dekel, one of my lab members, used TBC and expressed OR8 on a Xenophus egg, like I've shown before. And what you see over here is that that's the OR8, and those are the pulses. And every time you see that little arrow, means that Amir delivered octanol, and then you see a depolarization, which is that slope. So that's what happened when you use octanol, which is same as Jonathan, but when he mixed octanol with indole, labeled in the red line, you see a decrease in the current. And when he measured those two things, he saw that mixing indole with octanol reduces the current by 30%. So Amir did tons of pharmacological study, which I won't go through, but I just wanted to know that he did it. So that's where, sorry, as a conclusion, we see that indole inhibit OR8 function, similarly to DEET. Sorry. So my first goal, and that's where I come in, I'm the behavioral guy, was to understand if indole can inhibit short-range human host attraction. So based on previous results, we wanted to try it in an RNA cage assay. And what we did is we designed and 3D printed two-layer ring, which fits neatly in glove. In between, we put a protective net, so the mosquito could come and visit and smell the experimental hand, but they won't be able to bite the hand. So on the top ring, we put a chemical holder with a filter paper, which basically we pipe a different concentration of indole, and so the human odor and indole was mixed together. And after that, we put the hand inside the cage with 15 post-natal mosquitoes for 10 minutes. We shoot it, and this is how it looks like. So actually, on the left side, you can see the control. So the mosquitoes are coming and probing and trying to find a way to the skin. And on the right side, we see high concentration of indole, and we see much less mosquitoes. But we have the same number. So we measured 2 parameters, and we counted by hand. We just saw the videos and we just said, okay, there's a landing, there's a landing, there's a landing. So we count the number of mosquitoes on the region of interest, which is the net. And here you can see the average number of mosquitoes. And as you can see, we check variation of concentration of indole from 10 to the minus 6 to 1 molar. And we also checked dip in 1 molar as a positive valent control. And we see that as long as we have higher concentration, the mosquitoes come less to the screen, to the region of interest, and try to probe. And we also measured the average time on the net. So we basically count how many seconds they've been on the net when they landed. And over here, surprisingly, we didn't find any difference. And we thought that if they choose to land, despite the repellent, they just stay there more or less the same without any statistical difference. So the next thing we wanted to do, we saw that indole inhibits short-range human host attraction, but we also wanted to check long-range human host attraction. And for that, we use a wind tunnel. It's 1.5 meter long and 0.5 meter wide. And we actually put 10 mosquitoes inside the release chamber and let them acclimate for 1 hour. And after 1 hour, we open the release chamber and we open the odor source, which we use CO2 with octanol, and CO2 with octanol with indole. So the experiment was in complete darkness, and the trajectories was monitored through 2 IR sensitive light, infrared sensitive, two infrared sensitive cameras with infrared light. And over here, you can see an example of the output of the trajectories. Those who use MAK'IT may recognize it. So here we can see a bird's eye view of all the treatments. So we got a CO2, and octanol, and CO2 and octanol, and indole. And the color says the speed. So basically, the warmer the color, the higher the speed. So as you can see, we have slightly difference when we look at it by the eye between the CO2 and octanol. But when we add also indole, we see that the color changes dramatically. So we measured 3 parameters. The first parameter was like general speed, and here you can see the median and the statistical differences. And what you can see is when we elicit CO2 alone, we have a certain speed, but when we add octanol, the speed increases. But when we add indole to the mixture, we see a decrease in the speed. So that's about the speed. We also measured upwind velocity, which means velocity towards the odor source. And we see no difference between the CO2 and the octanol. But when we add indole, again, we see a difference in the velocity. And the last thing that we measured was tortuosity, which indicates for straight flight. So basically, 1 means straight flight, and as you go down, it means that they fly more tortuous. And overall, also, we didn't see any difference between CO2 alone and CO2 with octanol. But when we had the indole, again, we saw this. So we conclude from those experiments that indole inhibits short-range human host attraction. Indole inhibits flight speed and upwind velocity, and indole elicits tortuous flight. And okay, so we also wanted to see what happens with the larvae when we expose them to indole. So this is kind of a preliminary results. It's those experiments, we conducted them like last month. So I would like the video know. So what else should we do? So in general, the mosquito larvae expresses low odorant receptors compared to the adult stage. Their main ecological behavior is foraging and avoiding predation. So they have actually 66% of the odorant receptor that is larvae specific. It means that
expresses only in the larvae. And out of those 24 odorant receptors, we got OL2, which is indole sensitive odorant receptor. We got OL8, which is octanol odorant sensitive, and we got also OL9. So what's OL9? So Aedes aegypti uses different indole OL, which is OL9, to detect scatol. Okay, so here we see indole and scatol. So for indole, they use the same receptor, but for scatol, which is slightly different from indole. I mean, that's only the methyl group. So they use different odorant receptors. So both of them, indole and scatol, are hydrophobic and very volatile. And correlated with that, OL9 is 20 times more sensitive than OL8. So, as a continued research from what I showed you before, our first question was if indole can inhibit food foraging in the larvae. So what we did, we basically took 20 larvae and we starved them for 2 hours before each experiment. We took a crab food, this is what we feed the larvae, and we basically soak the crab food with water or indole, or water with scatol. And the food was placed inside a 3D printed igloo, which you can see over here. So we printed the igloo because of 2 reasons. The first one, that we didn't want the crab food to move a lot. And the second reason is that we wanted to create gradient of smell coming out of the igloo. So the water level actually don't overcome the igloo. Okay, so the larvae cannot move above it. So we place the larvae in one side and we monitor the whole event for 1 hour. And that's how it looks like. On the left side, we got the raw images. The raw video, sorry, you see the larvae is coming inside, eating some of the food and going outside. And we used image processing tools to monitor the number of larvae inside the region of interest, which is inside the igloo, so we can see the dynamic of them coming in and coming out. So we plotted the dynamic, and over here what you see is that's the time and that's the number of larvae inside the igloo. So that's the control, so we basically put nothing inside the region of interest, and as you can see we have very low numbers, and so I'm sorry that the index is a bit small. So there's almost no mosquitoes that are coming in, but you see sort of a trend of coming in, coming out. But when we put crab food inside, we see a huge difference in the dynamic. So they are coming in, they are basically eat, and they're coming out. So the first thing we wanted to check is how do they react if we will put the food crab inside the indole mixture. So with indole, with high concentration of indole, actually 10 millimolar, we don't see a difference, so the difference is really small between the food crab and the food crab plus the indole. So the trend changes a bit, but it's more or less the same. But when we put scatol with it, we see a different story. So we see that at the beginning, at the first 20 minutes, we see same dynamic, right? They go in and they go out. But after that, yeah, it's the scatol is labeled in green. We see that the trend changes and they don't come that much inside the igloo. So we wanted to understand or compare the different treatments in a 20 minutes time interval. So this is what we did. We took time between 0 to 20 minutes, between 20 minutes to 40 minutes, and between 40 minutes to 60 minutes. And here we see the dynamics. And what you can see is that food, and the crab food over here, it's pretty much consistent along this 1 hour. So they come more or less the same. With the indole, it changes a bit. In the first 20 minutes, we see a small difference, but I don't see it as I don't see biological meaning behind it because it's too similar, and it has no difference with the scatol. But in the middle 20 minutes, we see increase in the larvae number when scatol is presented. And we can also see it in the last 12 minutes. So from this project, until now, we conclude that indole does not inhibit larval foraging, food foraging, and it might even encourage them to come and eat. And scatol does inhibit larval food foraging. So of course, there's a lot to do. We want to test it with the variation of the concentration, maybe to do some GCMS in the water to see how the scatol is distributed. And that's it. I want to thank my lab members, my beloved PI Jonathan Lookwood, which has a birthday today, so happy birthday, Jonathan. I want to thank the University of Jerusalem and, of course, the ISF, which fund us. Thank you.
Thank you very much, and we have plenty of time for questions. So, Greg, yes.
Yeah, nice behavioral work. My question is that you mentioned that your colleague in the lab, when he did electrophysiology, there was only about a 30% decrement in the amplitude when he did the electric voltage experiments with ORA. Yet you had a really strong behavioral phenotype, but you also mentioned that the ORs in larvae are about 20 times sensitive. So I'm wondering if there's a higher affinity receptor in the adult, and maybe like OR8 is not really the the key receptor for the indole response. What are your thoughts about that?
So thank you for that question. Yeah, we actually thought about it. So we didn't use any knockout mosquitoes, right? So if you want to do a collaboration with us, you can do knockout. Please reach us. So yeah, of course, we can't conclude it. We see only correlation between pharmacological study and the behavioral study. But of course, we cannot conclude that that's because of the inhibition of ORA8. Yeah, maybe just circumstance. Yeah, thanks.
Yes, Marcel?
Yeah. Question about the duration of efficacy. Did you investigate time, protection time, also indole human, and the stability that this molecule could have if you put on the skin compared, for example, to DID or other reference?
Yeah. So no, actually, we didn't do it. We didn't check how long does it last. But as Potter said, volatile compounds evaporate in a short time. So we don't really know how long it will protect you, but we think that it would be less than DID, for example. Yeah. There was another question also?
Yeah. David? And then Oleg.
Thank you.
And then Marcelo.
I was wondering about the solubility of indole and scatol involved. Are they soluble or not?
Not that much. No.
If they are not, perhaps they stay in food, and you might not create this gradient. So larvae will go in, they will bite, and once they will bite, they might like it or not. But they will still be able to go in because water will prevent this to go far away.
So actually, when you take the patch of food crab,
yes.
So you soak it with the scatol and the water, and then you transfer it. And after you transfer it, after the experiment is finished and you open the incubator, it smells. Also scatol and the indole are high concentration. It smells like pieces. So we don't know. Because of this, we wanted to conduct a GCMS, right, to understand the distribution. If scatol is distributed, or does it stay, like you say, on the crop? Yeah.
Well, it was saying about Marcelo.
Okay. Thanks for that. Very nice. These are deposition cues for mosquitoes, right? Do you know how much scatol or indole there is in breeding cells? How are these concentrations connecting to what the female search for when it chooses the breeding cell?
Yeah. So no, actually we don't know that, but as far as we understand, indole is found in oviposition site, but within not that I remember about an article that proves that indole is connected for oviposition cues, right? So.
Yeah, I'm just thinking about the data that, two things, one was periodicity, and the other thing was that it seemed as though the scaffold only had an impact after they consumed, right? So might it be an internal ingestion kind of thing rather than a directional cue kind of thing? So I was curious about the periodicity.
Yeah, yeah. So what we observed, and which was very nice, I'll go to that now. It's over here, right?
Yeah.
So first of all, they go in and then they don't. And what we observed only by eyes, and that we need to measure, that would be a nice measurement to see how the crab food changes. I mean, do they eat it, do they not? Because if we put only crab food, the brown pixel will decrease. So it would be nice to do some imaging measuring of the food size, but they just touch it and they go away. They don't eat it.
Last question.
Yeah, I was just looking at that graph again. I will be very interested to see what will happen after that time point, because it looks like the curve is going up again with scaffold. So it will be interesting to see the duration of the effect, because it seems that it's inhibiting for about 40 minutes, and then it might go up again.
Right. Yeah, very nice. We should try that.
All right, thank you very much.
Hello everybody. First, I would like to thank the organizer for inviting me to participate in this event. Today, I'm going to present a part of the results of what was my PhD project. And this work is entitled Other Mediator Response of Gravity DCG type to Mosquito-Associated Symbiotic Bacteria. So, as we all already know, a DCG type is the mediator of several arboviruses, like those causing dengue, Zika, and chikungunya, and therefore represents a major threat to the health of millions of people annually due to their high preference for human hosts. Human environments and their surroundings provide suitable places for these mosquitoes to develop, because there is a variety of places where the mosquitoes can rest, permanent hosts, permanent feeding. One of the most important characteristics of these environments is that they have a wide variety of artificial containers that females can use as oviposition sites, and as a consequence, they will serve as larval habitats. But the capacity of the gravid females to exploit human-made containers depends on several key attributes of these females. So, finding and choosing an oviposition site involves a multimodal evaluation of several biotic and abiotic factors associated with the breeding sites. Among them, we have the presence of protein specifics for predators, salinity, pH, surrounding vegetation, organic matter. And we know that mosquitoes are polymetabolic insects. That means that they undergo a complete metamorphosis with immature forms and adults inhabiting very different environments. So, in this sense, there is a strong selection pressure on the oviposition site-seeking females, because the progeny ground, the survival, and the reproductive potential rely on their egg-laying decisions. As we know, this immature stage cannot move from poor quality habitats. So, mosquito larvae inhabit aquatic niches, and they acquire a significant proportion of their bacterial symbionts during this stage through feeding. But bacteria present in the larval habitats not only represent a food source for these larvae, but it can also play a main role in adult metabolisms, for example, assisting sugar and blood digestion. Moreover, bacterial communities associated with mosquitoes can also have impact the life history traits of these insects, for example, increasing their survival and longevity, also contributing to egg production, oviposition site choice, and with egg hatching. So Bacteria is a main component of these aquatic niches, and they promote abnormal larval development, assisting these in mature forms for energy storage and biosynthesis of vitamins. So with this, I would like to give you a whole picture of how these mosquito microbiota interactions can have an impact on the insect host. But now we will focus here in the oviposition. So several authors have already reported that volatiles from bacteria origin are capable to attract and elicit oviposition responses in different species of mosquitoes. And despite there is increasing evidence that bacteria can impact the behavior of the mosquitoes, little is known about what are the components that are involved in these oviposition decisions. So yesterday we already talked about the insecticide resistance. That is a big problem. So bacteria promoting egg laying represent a rich source of potentially attractive volatiles that could lead to the development of lures targeting the gravid mosquitoes. Then there are a wide variety of bacteria that is frequently reported associated with mosquitoes and also with the breeding sites. And one of them is Klebsiella. These bacteria have already been reported associated with Aedes anomalous and Culex mosquitoes. Moreover, there are reports that these bacteria are present in all the life stages of these insects, as well inhabiting different tissues like the midgut, the feces, and even the ovaries in Aedes aegypti. Which can give us an idea that maybe this bacteria could be transmitted by the gravid female to the breeding sites during the oviposition. And in agreement with this hypothesis, there is evidence using culture-dependent and culture-independent approaches that reported Klebsiella in artificial and natural breeding sites. So considering all of this, the means of this study was to evaluate whether the volatiles emitted by Klebsiella elicit oviposition in gravid Aedes aegypti. Moreover, we sought to identify the bioactive volatile organic compounds emitted by this bacteria and to determine the olfactory pathway by which these VOCs are detected. So for this, we developed a series of experiments. The first of them was a two-choice assay. For this, we use a backdoor cage that was adapted with two holes to let the tubing to go in and enter an oviposition cup. So this oviposition cup consisted of one cup that have inside, that have here in the wall a hole that let the tubing to go in. Then a second cup was stacked inside this one and have some perforation that will let the volatiles pass through it. A third cup that contains water, and finally a piece of filter paper that acts as substrate for the mosquitoes to oviposit. So we use this device in order to avoid any possible accident during the experiment, because we know that bacteria could be pathogenic for humans. So using an air pump, we generate an airflow that goes through two bottles, one containing the bacteria inoculated in tryptic soy broth, and the other sterile broth. We also control the flow that was going to the test cup and the control cup using a flow meter for each of the bottles. And we allow a single gravid female, we release a single gravid female 2 hours before the beginning of the scoto phase and allow them to oviposit during 24 hours. After this period of time, the females were removed and the number of eggs in each of the cups were counted. For this experiment, we tested 4 different bacterial loads against their respective control. That is what different concentration of the restricted soil rot. And here we can see what happened. So the VOC is emitted by Klebsiella induce a preference in oviposition decisions by Aedes aegypti in a bacterial load dependent manner. As we can see, the lower bacterial loads that were tested didn't induce any preference by the gravid females, but when we tried the higher bacterial loads, the mosquitoes had a preference for the cup that displayed these bacterial volatiles. So then, as we see that the Aedes aegypti prefer the oviposition cups mated with these odors, bacterial odors, we choose one of these conditions. In this case, was TSB 1% and this bacterial load to perform experiments, GC-EAD and GC-MS assays. So we collect the bacterial volatiles in Klebsiella culture using solid phase microextraction fibers. And here we have the results of the GC-EAD analysis. We evidence 9 consistent responses in all of the recordings that were performed. But of all of these compounds, just 2 of them were present in the bacterial cultures. The others were also present in the broth where the bacteria were cultured. By GC-MS analysis, we identify these 2 compounds as 2-ethyl-hexanol and 2,4-butyl-phenol. So, as we have evidence that the mosquito have a preference for these Klebsiella volatiles, we develop a synthetic blend using these 2 compounds and perform 2 choice behavioral assays using a device similar to this one, but avoiding all these theory that was not needed. Yes, Dantiana already mentioned that. So here we can see the results for the synthetic blend. We test this with 2 different mosquito strains, Rockefeller and Orlando strains of Aedes aegypti. And we tried 3 different doses of this synthetic blend. And we evidence that the mosquito also responds in a dose-dependent manner at only the highest concentration show a preference for the oviposition cup that contain the blend. Sorry, I forgot to mention that in the case of this experiment, the oviposition cups presented water inoculated with the synthetic blend or water, and the control water inoculated with hexane, that was the compound where the 2-ethyl-hexanol and 2,4-butyrphenol were preparated. So we also perform an experiment using a strain of orthomutans. This is mosquitoes lacking the odorant receptor co-receptor, and we didn't evidence any preference. So orthomutans were indifferent to the binary blend. And to confirm all these results, we also perform GC-EAD analysis using the synthetic blend, and we evidence again that the Rockefeller and Orlando strains respond to the 2 compounds, and there was lack of behavioral or physiological response in the orcomutants. So about the results that we have using the Klebsiella volatiles, this is in line with what was previously observed for good experience. That also some authors also report that this bacteria is attracted to this mosquito species, and they speculate that this bacteria could be inoculated from the gravid female to the breeding site when they are ovipositive. These authors also performed experiments to see if this have some consequence in the larval development. And in fact, Klebsiella can faster the molting from larva 1 to larva 2, which gives us an idea that this bacteria could be important for the development of these immature stages. So the conclusions of this work was that the headspace volatiles of Klebsiella cultures elicited egg laying in rabbit Aedes aegypti. The binary blend of these bioactive compounds identified in the Klebsiella headspace are detected by the odorant receptor pathway, and they were able to recapitulate the oviposition response of rabbit mosquitoes. And finally, this binary blend identified here has the potential to be used as a lure together with existing tools for mosquito monitoring and control. So I would like to thank all the co-authors involved in this work, Said and Betty, who performed the behavioral assays with the synthetic blend and the GCEAD recordings, respectively. Beatrix, who helped us with initial advice in the microbial component of the experiments. Sharon and Ricard that hosted me during the last year of my PhD and were great supervisors. Finally, Marcelo, that was my PhD advisor. Not less important, of course, the funding agencies, MAK'IT and FioCRUZ. Thank you.
Thank you very much, Nadia. Any questions? Carlo?
Could you please explain about the oviposition index and the behavioral answer?
Yeah, sure. So we counted the number of eggs that were collected in each of the cups, and then we use a formula where, for example, for the control was the control divided for the number of eggs collected in the control divided by the number of eggs collected in the blended control. And in that way, we calculate this oviposition choice index. And that was for the control, and the same, for example, for the test.
And this is with how many mosquitoes inside your, I'm sorry, I missed.
So each experiment have one single female, but we have at least 30 replicates for each of these different experiments.
Did you try to account for correct mosquito oviposition?
Not really. Like we just count the number of eggs and just provide these two different cups, one test and one control.
In terms of the oviposition, how much is it triggering the oviposition per se versus well or just attraction?
Sorry?
Is it primarily attracting the gravid females, or is it likely to be acting more locally, triggering the oviposition per se?
Could you repeat your question, please?
Is it primarily causing the gravid females to go to that location and then they do the oviposition, or do you think it's likely to be acting more locally? In terms of, do they visit both sites, but they lay more with this cue? Does that make sense?
Yes. I think that they are attract and also elicit the oviposition responses because there are also previous evidence that the bacteria can impact the oviposition response, for example, in Kulex, and they also developed this kind of experiment where they present like In that experiment, there were not only Klebsiella, but other controls, even other bacteria. The mosquitoes were able to be attracted by Klebsiella and also to lay more eggs in the oviposition cups that contain the bacteria.
Did you do experiment where you gave them a choice between the Klebsiella and the two odor blend to make sure that they were balanced? Because if they still prefer the Klebsiella, even though it looks like it's only two from this, and you only identify two from the GC. I was wondering if you did that control.
Sorry, I don't understand.
So, if indeed the two odor blend are only the two relevant odors from Klebsiella,
I mean, and in the conditions that we are testing the experiment, yes, there were the only two that we identified associated with the bacterial culture. But for example,
excuse me. So my question is, did you do a two choice between the Klebsiella and the two odor blend to see?
No, we didn't do that. So we don't know. Maybe.
University of a sandfly actually in the field. So sandflies are pretty tiny vectors, about 3 millimeters. They have a fairly global distribution in arid and tropical regions, and they transmit a variety of pathogens. The most significant one obviously are Leishmania, that are the agents of Leishmaniasis, but they transmit also Caron disease and some viral diseases. So in terms of the control, people have been using insecticide and repellent, but insecticides appear to be decreasing in efficacy and also impacting a non-target species. So I guess along the general theme of this meeting, that there is a need for more ecological friendly vector control approaches, and that's one of the type of things that we try to do in my lab. So by training, I'm an ecologist. So my belief is that by applying an ecological approach and trying to understand the ecology of the system that you're studying, you might be able to find ideas about potential weak links that could be targeted for the transmission, for breaking down the transmission cycle and the life cycle of the vectors. Sandflies are interesting because they have a completely terrestrial life cycle. The female, after she takes a blood meal from a host, often that's a mammalian host, she lays the eggs. And the oviposition site of sandflies is a big mystery, but generally they tend to lay eggs in sheltered places, dark places that are rich with organic matter, and animal burrows and dens tend to be a common oviposition site. So the life cycle of a sandfly is fairly slow compared with mosquitoes. It takes about 5 weeks to complete an entire life cycle. So they go through 4 instar stages, and then they molt into pupa, and then eclose into adult. So when talking about kind of generalizing about the life cycles of sandflies across the world, we could, and their oviposition sites, can generalize it to, so again, there are many sandflies that are what we referred to are tightly coupled with their host, in the sense that they often don't only take blood meal from a certain host, they also like to live in the burrow of that host. So with the rodents, we often see situations where the sandflies also breed and shelters in animal burrows. So we call this syndrome, we call that tightly coupled vector host system. And that's and that occurs with many sandfly systems. But there's also the more general kind of vector situation, so like mosquitoes, where the vector is independent in terms of its breeding of the oviposition site of the host. So we term this syndrome uncoupled vector host systems. And actually, we applied some simulation modeling to try to evaluate what is the consequence of the degree of dependency of the vector on the host in terms of the disease dynamics. And as you can see in the lower screen, there is quite substantial difference in the outcome of a theoretical disease with generally a negative impact of host density on the final proportion of on the prevalence, whereas where we have positive coupling, tightly coupled situation, then actually host density can actually increase the final prevalence in the host population. Yeah, so that's kind of a new idea, and we're working on that, and hopefully publish it soon. So I've been working on the system of Phlebotomus papatazi and gerbiline rodent hosts for quite some time, and these are a system that could be referred to as tightly coupled, because the sandflies, they live in the burrow and breed there. So again, so when looking at tightly coupled vector host systems, types of approaches that could be applicable here are things like systemic control, where you target the host, where you feed it with a systemic agent that affects the sandflies, but not the host. And another approach is oviposition site detection, and basically oviposition attractants. Basically trying to mimic the type of cues that attract gravid females to an oviposition site, and potentially into a lethal ovitrap. Oops. Yeah, so again, so this is the model system that we've been working with for Phlebotomus papatazi. And again, one of its preferred rodent host is Psammomis obesus. So that kind of puts us back to the general framework of many of what the speakers here were talking about. Basically, what constitute a good oviposition sites. And for many insect vectors, obviously, they don't have maternal caring. So basically, the most important choice that the mother would do is the oviposition site. So the mother needs to think very, very carefully about where to lay her eggs. Basically, the working hypothesis that many people have used is the preference performance hypothesis, that the female's oviposition site preference should correspond with the performance, the growth and development of the larva. And in that sense, females are expected to have evolved the ability to respond and identify cues that are reliable indicators of suitable and high quality of oviposition sites. Yeah, so some of my work focused on olfactory as well as visual cues that guide the optimal oviposition site selection of Phlebotomus papatazi. Yeah, so oviposition attractants are a useful target, have the potential to be useful target for control, because they target gravid female, which are the physiologically later stage in the life cycle. And obviously they have blood fed at least once, so they have higher potential of being infected. And also if you intercept them before they lay the eggs, then you are making an impact on decreasing the population growth of your vector population. Yeah, so again, so when making a oviposition site selection decision, female are expected to look for places where there's food for the larva, where there's no predators or high competition, and in general suitable abiotic conditions. Yeah, so the type of methodology that we've been using is kind of combination. So again, so this is a collaborative work between me and my colleagues, Kobi Shal and Logu Punasami at the North Carolina State University. So I'm doing the, I have sandfly colony, and I'm doing the behavioral bioassays that include olfactometers of different types, either a linear olfactometer. We have like free flight cages with sticky traps, and we have oviposition bioassays where actually so in the attraction bioassays, we don't allow the females to lay eggs, but we just look at their orientation with putting a stimulus in one cup, putting a control in the other cup, and basically seeing how many females are intercepted in a sticky mash that we put in the cage. In oviposition bioassays, we actually, again, count the number of eggs that are laid in the stimulus containing cup versus the control. And we also do some field experiments with our preferred field site actually being Israel. Yeah, so we include, so again, my Logu Punasami, my microbiologist collaborator, is doing the identification and culturing of bacteria and of fungi, and in Kobi Shal's lab, he's doing basically GCEAD, basically identifying the components of the source material that induces antennal response, which then gives us a subset of candidate compounds that then could be tested in the lab with respect to their behavioral effect. So with respect to organic matter and microbes, we kind of started our work by comparing the attraction of gravid females to different stages of the rearing media of the sandfly. So again, the sandflies are feeding on organic matter, and we're using kind of a combination of fermented rabbit chow and rabbit feces. So we gave them so we used a set of two-choice attraction experiments where we compared the attraction to rabbit feces, fresh larval food, and then second, third means that this is a medium that have been used by larvae for a while, and mainly contains basically feeding and defecating larvae. Then a medium that had fourth and pupa, and also an old medium from which all the adults have already reclosed and died. So in this figure here, you can see that the most preferred oviposition medium is the one that have been what we refer to as the larval condition medium. Basically, this is the larvae medium that contain actively feeding and defecating medium. So again, so they really like that. And we followed this experiment by decoupling the aging versus the larval presence, and indeed show that the females prefer the larval condition medium over a medium that was aged the same amount of time but didn't have larvae in them. Yeah. So this raised the hypothesis that that larval-induced microbial community changes the attractancy of the medium. And potentially this attractance also has an effect on the development of the larvae. So to test the effect of larval conditioning on the structure of the microbiota, basically we did a microbiota molecular analysis where we took where we did a series of food that starts from fresh, and then one set had larvae in it, one set did not, and we basically took samples all through the development stage, different development stages of the larvae until they have eclosed. And so this is the microbiota of the age medium, the one without larvae, and this is the microbiota structure of the larval conditioned medium. And these are significantly different. But what is really interesting is that they are very different at the earliest stages, week 2 and week 4, where the larvae are present in the medium. But then in week 4, in week 6 and week 9, where the larvae have already closed, then the microbiota community structure appears to actually converge and become more similar than previously. So suggesting that indeed the presence of the larvae changes the microhabitat of the microbes in a way that creates different microbial community, and the different microbial community emit a different volatile plume that makes one source more attractive than the other. And this is one of the parts that we're still missing, that we'll be studying next, the different volatiles that are emitted from the different microbiota structure. We also evaluated the development, whether larvae indeed need bacteria in the medium by, again, growing them in sterile versus a non-sterile medium. And we also grew them in medium of the different stages, including the one that is supposed to be most attractive, the second, third. And so with respect to sterility, indeed, larvae grew worst in the sterile medium, suggesting that indeed bacteria are important. But surprisingly, they grew also worst in the medium that was the most preferred one. That was surprising, and that suggests that mothers don't necessarily always know best. So they knew they did the right decision with respect to importance of bacteria, but not really with respect to the second, third. So that's an interesting result. And I need to rush a little bit. Yeah, so indeed I isolated several bacteria, and we identified some of them that were super attractive to the sandflies, and especially this one, the SSI9, was super attractive. And then yeah, so we followed that with a GCEAD analysis to try to evaluate what is in the volatile medium of this very attractive bacteria. And we found 4 that were unique to this bacteria when comparing the microbiota and the EAD profile of that bacteria with others that were less attractive or non-attractive. So one of those compounds, 3-methylbutanoic acid, also known as isovaleric acid, we ran several attraction and oviposition bioassays with that, and show that actually it is it is both attractive and also inducing oviposition. And this graph also shows geosmin, but that was attractive, but I'm going to skip that. Yeah, I'm kind of running short on time. So I think I'm going to skip the part about attraction to conspecifics, because I want to get to the time to talk about visual cues, but I'm going to mention that sandflies were attracted to eggs and first instars, and we showed that this attraction is mediated by dodecanoic acid. I'll just have to kind of run quickly by that. So again, this is the dose response effect of the dodecanoic acid. So what we're thinking is that the dodecanoic acid, because it's a larger molecule, probably is more of a position stimulant, whereas isovaleric acid acts both as an attractant on a larger scale, but and also can act as a oviposition stimulant. Finally, I want to talk about the visual cues and the photoperiod of sandflies. Again, so as I mentioned, sandflies live in often, at least in the old world, they live in arid areas, and in arid areas they live in animal burrows. So they're expected to visually, one would be expected they would be attracted to all kind of cues that similar to entrance of animal burrows. And being nocturnal and crepuscular, they should also they're expected to lay eggs at night, but we don't know exactly when. So this is the setting of the experiment, where we presented gravid sandflies with oviposition cups that contain organic matter in both cups. The only difference was that one was covered by a black paper and the other one was clear, or it was covered by a white paper and the other one's clear, or we also had some experiments of white versus black. And in the one that we studied the ideal periodicity, we had a single cup in the middle of the cage that we replaced every 2 hours, and we just counted either the number of sandflies that were caught or the number of eggs that were laid. So with respect to attraction, we saw that was a peak of a position site search activity actually in the later part of the night, like between 3 and 6 at the stage of the scotophase, but oviposition actually occurs at the earlier part of the night. We also showed that highest preference was corresponded to the time where they were mostly active in terms of searching. So that's very interesting. In terms of the visual cues, the gravid females appear to be highly attracted to dark cups, and over clear cups, or dark cups over white cups. They did not prefer white cup over a clear cup. The color did not affect the oviposition, only affected the attraction. And most interesting here was the fact that when we compared gravid females with other stages, basically non-gravid females or males, this visual stimulation applied only to gravid females. It did not affect other stages. So it's only that gravid females are the ones that are attracted to black cups. I think I'll skip this one for lack of time, and I'll just summarize my talk with respect to the type of application of oviposition, attractants, and visual cues. So the combination of, again, identifying the compounds and potential bacteria that could act as, again, as attractants. First, it's very important to understand the biology of the oviposition, biology of sandflies, but this could be translated for the development of things like lure that could be used for surveillance, and that lure could also be applied to an oviposition, to a lethal oviposition trap that could be used for the control and mitigation of sandfly-borne diseases. So I think that's all I have, and this is the team that I've been working with. Again, the NC State team with Kobe Schall, Charles Apperson, and Logo Punasami, and this is my team. And I thank the NIH and the North Carolina Technological Center for the funding. Okay, that's it. Thank you very much. I hope that I was.
That's all right, Gideon. It's okay. We have time for questions. So. Yeah.
I have a question.
Yeah.
Did you try this experiment with the fungi? Which kind of fungi can attract the female? Did you try that?
It's hard for me to hear the question. Can Marcelo, can you repeat the question?
I don't think I can repeat it perfectly.
About.
Just a second.
Can you hear me?
Now this is better, yeah.
Hello. Yes, I had a question about the experiment that you did with the bacteria. Did you try with the fungi? If there is some fungi that can attract the female?
Yeah, very good question. Yes, actually we just started working with fungi, and we actually were able to isolate from the same medium, the attractive medium. We were able to isolate 10 fungal strains, and my colleague Logu made them into like this kind of like agar plugs, and I ran an experiment with them, and we actually found that they were highly attractive as well. So the community of the fungi was attractive to the sandflies, and indeed one of the next phases of our study will be to basically repeat the same approach of what we did with bacteria and to try to identify the specific fungi that are responsible for this attractive effect. Thank you for the question.
Thank you.
Question? No question? I will ask one. Can you hear me, Gideon?
Yes, I can.
Yeah, I was wondering whether maybe you addressed that already and I missed it. And if it's the case, I apologize. Do they have a bed hedging oviposition strategy, those flies, or do they just lay their entire batch at once at one specific location?
My goodness, the question that you guys are asking me it's as if I ask you to ask me those questions. So yeah, exactly. They do, and they employ both spatial bed hedge. I mean, they appear to employ both spatial bed hedging as well as temporal bed hedging. There was a spatial bed hedging. Again, we don't know in the field, but in the lab, I ran experiments where with a oviposition experiments with a single female, where I gave her several sites of equal or different quality, and I showed that indeed the females, a single female, can in a way skip oviposit and lay her entire, basically distribute her batch of eggs among different oviposition sites, and their relative amount corresponds to their quality of the habitat with the amount of food that I gave them. With respect to temporal bed hedging, I showed that that organic matter, basically of aquatic extract of organic matter, can determine the hatching time of egg. So basically suggesting that the larvae that are still inside the egg can regulate their hatching time according to some cues of the conditions that are outside. So again, this is preliminary, but these are interesting results. I hope that answers the question.
Absolutely fascinating. Thank you.
Thank you.
Great. Other questions? Yeah. In the chat, Gideon.
In the chat?
Yeah, I'm trying to.
In the chat.
Is it possible to incorporate the GPF protein in the eggshell to help try?
It's possible to incorporate.
Try and find where eggs are laid in natural environments.
Gideon, did you?
Yeah. I'm there, but I'm just trying to wrap my head around. Yeah, that's a good question. I need to think about it somewhere. I cannot answer that off the cuff. I'm sorry. So yeah, or I'll be more than glad. Maybe you can email me the question and I can think about it some more. GPF protein. Okay. Okay.
Yes, David.
Hello, thank you for being there. Just a question. Some of these parasites they transmit, they are transmitted vertically. Is that so?
The Leishmania are not transferred vertically.
Is there any that is transmitted vertically?
Virus?
The viruses, I might possibly. I'm not quite sure about the viruses. But one thing that we're still trying to study is about the gut microbes. If the gut microbes are transferred, if there are some gut microbes that are transferred vertically, we don't know that yet.
But my question was like if they are transmitted vertically or whatever, or the question is like, do infected females would change the reproduction selection with what?
Yeah. So if infection could affect the gravid oviposition behavior.
Yeah.
Agenor Mafra-Neto. Agenor is the president of ISCA Technologies, a company dedicated to controlling pests through developing behavior-based control tools. And he will speak about the development of commercially viable semiochemical tools and strategies. Agenor, are you available?
I'm available. Can you see the screen or not yet?
Yes, we can.
And you can hear me?
Yeah, perfectly.
Amazing. Okay, technology is amazing. I wish I was there. Okay, everyone. So we're developing all these different things. So how can we bring them to become commercial and therefore being used in the field to control all these different vectors of diseases? Here today, ISCA Technologies is a company that started in the ag, and we also venture into the vector control area. So I'm going to talk about developments that we did, both in ag and in vector control. And one of the things that is really interesting is that many of the insects that you commonly see in the field in agriculture also transmit diseases. They transmit fungi, bacteria, and viruses, depending on the species. So most of them they vector diseases either actively or passively. So let's talk about the development of commercially viable semiochemical tools and strategies now. Here we go. Conventional control is based on this, like how to best penetrate the crop and bring insecticide everywhere in that crop in order to kill the insects that are around this field. So it's you put a lot of insecticide, you're always chasing the pest, you're bringing this insecticide everywhere, including the soil and every part of the plant. Technologies for blanket insecticide spray, the new technologies are usually how to penetrate this canopy, how to envelop every single part of the plant in order to protect these plants. ISCA has a different approach. We believe in semiochemicals and evolution. So that's a good thing. So the deal here in America, that's a very innovative thing, to believe in evolution and so we believe in semiochemicals, so we develop attract and kill systems. What attract and kill systems do, you basically put a little drip of your formulation in the field, and the pest comes and chases the insecticide. We add a tiny little amounts of insecticide, is basically 1% or less of what the grower would use in one application. And now we have a formulation that sits there for 15 days or a month, attracting and killing insects. I'm going to show you, and then the beauty of this is that you apply this, so it's basically half a liter per hectare. So you put this little half a liter over 100 meters in this little line, and then you skip 100 meters, leave that crop completely untouched by insecticide, and then you do the other line. So this, what I'm going to show you next is what happens under this line here in the field. You walk 2 meters to one side or 2 meters to the other side, you don't see this event. So here, you're going to see like this white things here. This is the application of Noctovi, an attractant for Lepidoptera. And you see all these different moths feeding on the formulation. This is like 30 minutes after the application. Here is an hour or 2 hours later. As you can see, the moth here, there are sometimes so many moths that the plant actually bends and then you walk 2 meters to the right or to the left, you don't see moth. So what you find in the morning under those lines is like lots of dead moths, and growers like to see dead insects, so you end up having a successful product. The other reason for the success is that there is an increased yield and increased profit. Sometimes we can double the grower's EBITDA with 3 or 4 applications of this product in the field. Another thing that is interesting is that when you are dealing with large row crops, and so 10,000 hectares of row crops, you have to use a tanker with 50,000 liters of insecticide in the conventional control, whereas only a barrel or 200 liters of insecticide using attract and kill. Over the years, since 2014, when we launched Noctovi, we have eliminated about 10 million kilos of conventional pesticide. So it's good for the grower, it's good for the environment, and it's definitely good for my company. The other, so stink bugs. Stink bugs are, they are not controlled by genetics or by, for example, GMO crops and things like that. So they have been escaping the escaping control in the field, and they became extremely resistant to many different insecticides. So stink bugs became like a group of pests that are increasing the damage in the field, and they are accumulating from one crop to the next. When we have situations like in Brazil, where you grow soybeans, corn, cotton in the same field throughout the year, you end up with extremely large populations of these insects. They cause tremendous damage. Some of the damage is physical, but a lot of it is the damage is transmission of pathogens in the field. So you end up with healthy seed, which is what the grower wants, all the way down to severe injury. And the funny thing is that usually when you harvest, you don't see problems with the seed so much. So this is like one example of a trial where we did different types of different doses of our formulation in against the control, the farm management. Just to show you here is the same thing. We just put that line in the field. And here in this case, we jump 30 meters and then we do another one. These bugs don't move as much as Lepidoptera. And here is what happens to a stink bug. They are attracted to the point source. Then when they touch the point source that induces phagostimulation, they start feeding. And depending on the insecticide that you use, here we use metomil, which is an insecticide. If the bug breathes the label, they die. And they feed on the formulation, and then they fall into the ground and they die. This is this video is like a fast video of about 2 minutes. So you end up having that in the field. So you reduce the population when you in the field when you use actor stink bug, and you in the control you have significantly more pest. When we look at the samples of soybeans collected to estimate yield, first we find that the damage is significantly lower where we use actor stink bug, and the yield is significantly higher. So what we get here is that we get a more effective, safer, less resistance and low cost product to go to the field and protect crops. Can we bring this to mosquito management? So one of the examples that I have is Vectrax, which is a nectar-based attract and kill system where we have feeding stimulants, attractants, and we add insecticide. And the mosquitoes are attracted the same way as though you saw those moths or stink bugs in the field. We did test development of this whole product in greenhouses in Tanzania, where we put Vectrax on top of this roof, release the mosquitoes inside these greenhouses, basically 500 mosquitoes, and the only source of Vectrax and insecticide is up here on top of this roof. And then we put someone inside that hut to measure the number of mosquitoes landing. And then we find out that basically in the first night, you get 400 landings in the control, very less than 50 landings on the Vectrax side. The next night, all the mosquitoes in the Vectrax side are completely dead, and you only have landing on the control area. After doing a lot of field lab and greenhouse trials, we end up going to the field where we selected 8 villages, about 500 houses in the Vectrax treatment, 400 houses in the control villages. We put CDC traps in the houses in the villages, check the mosquitoes, and that way we monitor the population. During these 9 weeks of pre-treatment of both Vectrax and the control villages, we end up having more or less the same dynamics, the same population. Then we go to the field. At week 9, we started the treatment. The treatment consisted of spraying Vectrax at the eaves of the houses. The idea is that we would stop the mosquito from going inside the house to get a blood meal. And what we seen was that in the Vectrax, the population continued to decrease, whereas in the houses, the control, you end up having a tremendously high peak with that, and the mosquitoes were biting on people. When we add that to Vectrax together with larvicidal splat bag that attracts and kills females, you end up, female mosquitoes, sorry. You end up with less malaria transmission in the Vectrex side than in the control. So in conclusion, Vectrex can be used, basically diverse and kills blood-seeking malaria mosquitoes, attracts and kills malaria mosquitoes in real-world situations, and when used with a larvicidal, it reduces malaria incidence. So again, the same thing, more effective, safer, no resistance, and low cost. Now, with mosquitoes, you end up having, people use repellents a lot. I was wondering, can we use repellents in ag? The problem is that the repellents, when you're protecting a person, these repellents cost a lot. So can we bring them to ag and still maintain that low cost? I'm going to talk about repellents for beetles, CBB Repel, Splat Verb, and Beetle Guard. Scutellidi beetles, coffee bean borer, mountain pine beetle, and ambrosia beetles, they all are symbiotic with fungi, and they the death of the tree, in especially mountain pine beetles and ambrosia beetle, is related to pathogenic fungi. This here is a forest that should be completely green and is due to the attack of mountain pine beetle. Each one of these red trees is dead. So there are huge number of trees that are dying here in the US. In collaboration, Steve Munson and Chris Fettig, we developed Splat Verb, which is you put like 4 dollops of splat into on the tree at the chest height, more or less, and then you basically control, you protect that tree from the attack of these beetles. When you don't use that, you end up with 93% of the trees dead. We repeated this for several years, and is the same. We get more or less the same statistics. The other thing is that there is a halo effect to these repellents for these beetles. And with Splat Verb, if you measure a diameter of 10 meters around that tree, you end up with 0 dead trees, whereas in the control, you end up with 61 trees on average, 2 kills per sample unit. We also found out that the dose that we use can be reduced, and the product still maintains the effectiveness. When we tested this in area wide, we find out that Splat Verb is able to reduce, in contrast with the control, 84% these mass attacks of this beetle. So it can be used in area wide. So Splat Verb protects single trees 100%. It creates this halo effect. And it suppresses 84%, which is almost as good as using drenching these trees with insecticides. When we look at the other ambrosia beetles, these ambrosia beetles are tremendously, they are beetles that are usually symbiotic with fungi. When we use these formulations, Beetle Guard, a repellent, with the red ambrosia beetle, it's a beetle that is killing red bay in the US, basically eliminated red bay from the southern parts of the United States. You see that when you use the formulation, you end up with a much lower number of attacks in these beetles attracted to these trees. When we simulate a tree with bolts for another ambrosia beetle, we find that with Beetle Guard, we end up having a much lower level of attack of these simulated trees than in the control. So the next is like the CBB Repel, which is it controls the coffee bean borer. Coffee bean borer is causing a huge damage to coffee. Everybody drinks coffee. I don't know people that are really nice that don't drink coffee. So this is very important to all of us. And it can cause 80% of damage in the untreated areas, and it's found in at least 50 countries. The insecticides that are being used is not working, and people are having trouble, especially with the global environmental changes that are happening. I believe in those, even though I'm in the United States, that there's climate change. But nothing seems to work. So I had to go to Hawaii and work with Mark Wright with the CBB Repel in order to figure out how to protect this. And we found out that with CBB Repel, we are able to reduce tremendously the infestation of berries that are in this high infestation areas. Now, in conclusion, so we have seen that semiochemical tools work for the management of vectors of diseases. So it works for ag and human vector management. They are usually more effective than conventional. That allows a company like ours to sell our products, even though people want to use insecticides. It's safer than insecticides and is amenable to resistant management. And we have been able to do that at a low cost, same cost as an application of insecticide. And these are some of the products that we talked here. The deal here is that to get mass adoption of this, we need to have a very powerful value proposition. We have to show that our products are efficacious and they don't cost more than insecticides. They have more advantages than that. So our products that we end up commercializing have to have a better performance than conventional insecticides, and they have to cost the same amount as an insecticide. And that's a very tough barrier to get, like $5 per hectare. So we need to be between $5 and $40 per hectare. So that's the amount of money that you have to create a product that can be used in the field and sold, and everybody wins there. This has been a collaboration with many people and with the support of many institutions. And man, I wish I was there. Thank you. Okay, no questions. Thank you.
Thank you so much, Agenor. I think we have time for a couple of questions from the audience. Yes, great.
Yeah, what is the molecular target for the Vectrex?
Sorry, can you repeat that?
Yeah, for many insecticides, they're voltage-gated sodium channels that are the targets. What's the target for the killing of some of these products like Vectrex?
I don't know if I understood the insecticide that we use, Vectrex.
Yeah.
We can use spinosad, but we can use several other insecticides, including insecticides that are for which the mosquitoes are resistant to, like pyrethroids, because the dose and the mode of action, like these insects are touching, so you get contact, and they are also feeding in the formulation, so you end up with different modes of action there. And the fact that they're feeding in these formulations, you can overcome resistance, for example. But the fact that they're also touching and feeding allows you to go with insecticides that are, they are organic or they are less effective than others, allowing you to less effective if you are to spray that in the field. But because they are feeding on it, you end up killing the insect, even though, and sometimes with very, very mild insecticides. The same thing goes for the Lepidoptera and for the stink bugs.
Okay, thanks.
Nelson. They are more or less a question about composition of the drugs and what they put in it in terms of insecticide, but also in terms of attractants. Is it the same for all the kind of species they target, or algae, or behavior, or how they can mix this together? And it's a generic with product, or it's really specified according to the target the species they target.
Did you hear it?
It was very difficult to hear, but it has something to do with the composition of Vectrex and the composition of species that you attract.
Yeah, actually, whether, besides the insecticide part, if it's different for the different targets, like different insects, or if it's more or less the same composition, and how do you manage the composition? Okay, so the composition is really important because you can make this the composition of these attractants very species specific or genera specific. And so the more components you have in your blend, the more specific you can make it. But for us, it's important to have a level of the product has to be general enough in order for us to have a good market and have a good penetration in the market. So usually our mosquito attractants, they have been selected. The blends have been selected to attract the Culex, malaria mosquitoes, the Anopheles and Aedes, in order for us to be able to go to the field. Another thing that is important is that you cannot attract mosquitoes and kill bees. So you need to be a little bit selective on the product that you're on the blend that you're using, so you don't attract bees. Usually we use repellents for these known targets. Any other questions? Yes, Niels?
Yes, I know in some of your applications, there's still a very high selection pressure. Did you see, for example, with your moths, any behavioral resistance?
Not yet, because these solutions are being brought in as part of an integrated pest management. So it goes into a program that the grower is continue to use insecticides for different pests plus our product. So our product basically alleviates a little bit the pressure that natural enemies and other known targets have in the field. But so far we have not seen that like an evolution of resistance or anything like that. But it's possible that will happen, but we haven't seen it yet.
Okay. Do we have any questions? Okay. So I think we have to keep on with the schedule. Thank you very much, Agenor.